Heartily welcome all the dignitaries for the online vocational training on immunological advancement in male and female fertility under the Institutional Development Plan (ICR) organized by Department of Veterinary and Physiology.
અવાજ એટલો જ આવશે
એ લોકો એટલા જ
Program at first we start with our university song.
આખો હોલ બનાવ્યો છે પ્રિન્સિપાલમાં અંદર જવાનો તો દિવાલો તો પાર્ટીશન વાળીને હોલ બનાવ્યો છે આના માટે હું કાલે ગયો તો પરમ
University song છે
Sound is not coming properly.
Thank you, everybody. With the sweet and melodious music song, I would like to request Dr. P. Taksar, Principal and Dean of Veterinary College, Junagadh, University, Junagadh, to have a welcome address, sir. Please.
Namaskar. The Junagadh Agriculture University, Junagadh, under the aegis of Institutional Development Project, sanctioned by ICR and funded by World Bank and the state government, has various components, and under such component we have today a virtual online 5 days vocational training on immunofertility problems in male and female. On this occasion of the inaugural function, we are having our Honorable Vice Chancellor and Director of Research and Dean PG Studies and Principal Investigator of this very project, Institutional Development Project in Junagadh Agricultural University, as President of the function. Sir, on behalf of University and on behalf of faculty and team IDP, I welcome you to this inaugural function, and with your permission, we have arranged so far 10 such vocational training since inception, and this is the 11th on very prominent area, that is animal reproduction, on which the production is ascertained. We have with us today a very newly designated and very first function of this university level, that Dr. Gajipara is from today onwards as Director of Extension Education, and he is with us. I welcome you, sir, to this inaugural function. After a long time, we have been requesting him as a professor and planning officer, but we are very happy to see you with us today in inaugural function as Director of Extension. I welcome you to this auspicious inaugural function of vocational training. We have with us two co-PI of the project and Dean of the faculty, Dr. Narendra Kumar Bhotia from Agriculture, Engineering and Technology faculty. I welcome you, sir, as a co-PI also and as Dean of the faculty. We have with us Principal and Dean of the Agribusiness Management, Principal Agriculture and Horticulture faculty, and co-PI of this project, Dr. K. Kut with us. I welcome you, sir, to this inaugural function. We have with us the Director of Student Welfare, Dr. Malam, who is also a co-PI for student extracurricular activities pertinent to university as well as institutional development project. I welcome you, sir, to this function. The key personality of the ICR and IDP nodal officer, Dr. Pramod Mohanat, is with us to witness the activities as well as to look after the whole vocational trainings and related component of IDP. Is also today with us to witness the inaugural function. I welcome you, sir, to this function. The organizing secretary and associate professor and head of Department of Gynecology and Obstetrics, Dr. Vada, and the co-organizing secretary as well as convener of IDP cell of this faculty, Dr. Arun Kumar Sharma. The team of this organizing vocational training, various professors, the research assistant, research associate related to IDP project, my subordinate staff, and those who are directly and indirectly involved, the media person, the staff from the Director Extension Education for making documentation of this whole training, are all with us. I welcome one and all to this inaugural function of Institutional Development Project Vocational Training. Sir, in this training online, we have with us a versatile personality as a chief guest, Dr. Madan, who is former Vice Chancellor of Mathura Veterinary University, former DTG at ICR, and today he is with us to act not only as the chief guest but also to bless with his vast experience in subsequent session as a lecture on immunofertility in male animals and females. Equally, a very experienced personality from our own state, Dr. F.S. Kavani from Anand, who is earlier Dean of the Veterinary College Anand and the former professor and head and present visiting faculty to Junagadh Agricultural University, has joined online as chief guest of this function. I welcome both these professionals at this very experienced age has given consent to bless our younger professionals. Comparatively, the younger college in India, Junagadh Veterinary College, and the faculty related to animal production and reproduction. I welcome you both, sir, on behalf of the Junagadh Agricultural University, College of Veterinary Science, and the Faculty of Gynecology and Physiology together. We have with us many invited guest lecturers who are subsequently to deliver their expertise lectures from STAU, Dr. Suthar, from AAU, Dr. Panchal, Dr. Dhami, and from Ludhiana, Hisar, IVRA, Ijatnagar, and Maharashtra, from Hapsu. They have also joined this inaugural function online. I welcome all these experts in the field of animal reproduction, animal physiology, and the animal production overall. I welcome once again all the participants of my own college, so also the faculty members who have registered this very useful vocational training online. To the best of my knowledge, yesterday up to 5 o'clock, around 750 registrations online have been achieved, which includes the UG and PG students of Veterinary College Junagadh, my own faculty members from the Junagadh, and some of the invited, and many are listening and watching online by link on YouTube and Facebook. So the new very opportunity to conserve, preserve, and to progress, the online platform has been very rightly utilized, though with little constraints. We also have this first meeting of this kind online in this particular place. And again, I welcome one and all under the presidency of our Honorable Vice Chancellor. Thank you.
Thank you very much, sir, for the welcome address to all our online and offline dignitaries. Now it's time to welcome the dignitaries which are available at the institute. We would like to have welcome with the flower flowers. So first, I would like to invite Dr. V. Chancellor to welcome Dr. Honorable Vice Chancellor Dr. Nikki Chovakia, sir. Please. Thank you, sir. Now I would like to invite Dr. Vicky Gog, sir, to welcome Dr. H.M. Gajipara, Director of Extension and Education, Junagadh Agricultural University. Thank you very much, sir. Now I would like to invite Dr. A.K. Sharma to welcome Principal and Dean, Dr. N.K. Gotia, sir. Thank you very much, sir. Now, Arjun Odeja, Dr. Arjun Odeja will welcome Dr. K. Poon, sir, Principal of ATM and India Principal of Agriculture College of Junagadh. Now, I would like to invite Dr. Akshay Baria to welcome Dr. V.R. Alam, sir, DSW, Junagadh Agricultural University, Junagadh. Thank you, sir. Now I would like to invite Dr. K.T. Wala, Organizing Secretary, to welcome our Principal and Dean of this college, Dr. P.A. Khan, sir. Thank you very much, sir. Now I would like to invite Dr. V.K. Singh to welcome Dr. P. Nanon, sir, ADR and co-PI of IDP program. Thank you very much, sir. Now I would like to invite Dr. D.M. Borkataria, Assistant Professor, Gynecology, to welcome our Organizing Secretary, Dr. K.T. Wala, Associate Professor and Head of Gynecology Department. Thank you very much, sir. I would like to invite Dr. Akshay to welcome Co-Organizing Secretary of this event, Dr. A.K. Sharma, sir. With this now welcome, now we inaugurate our program with the lighting of the lamp. So, all the dignitaries to have a lighting the lamp and open this online vocational training on immunological and advancement in male and female fertility, which is shooting under the IDP plant ICR New Delhi.
By the time a bit interruption, I would like to introduce Junagadh Agricultural University to the personalities, those who have blessed us as chief guest and those who are online. The Junagadh Agricultural University is one of the state agricultural university out of the 4 in the Gujarat state. It is in the western corner of the Gujarat, which includes 10 districts of the Saurashtra region. The Junagadh Agriculture University has got the Faculty of Agriculture, Horticulture, Agriculture Engineering and Technology, Fishery Science, Home Science, Veterinary Science, and many allied subjects in which it offers the diploma and certificate and vocational training course to the stakeholders. The very animal science component in JAU is because we all know that the Gir is the gold of the state and Gir cow is the gold of the country, which has given name to the India abroad also. We have with us 100-year-old cattle breeding farm at Junagadh Agriculture University, which shelters almost high potential genome of the genome of the Gir animals, around 650 Gir cows and 350 equally potential Jafrabadi buffaloes are with us. The veterinary college, born from the womb of the cattle breeding farm in 2008, and this is the 11th year of its starting. Over a period, it has achieved many milestones, and now it is recognized by Veterinary Council of India since its inception and accredited by the ICAR New Delhi. The faculty also has one diploma course under the Polytechnic in Animal Husbandry. After 10th, it is 3 years course. Likewise, 5.5 years MSc scheduled, VCI recognized BBSc and AH course. So also the MBSc and PhD. We have several research schemes under PLAN, ICR and other agencies. We also have a full-fledged teaching veterinary clinical complex where almost 45 on an average OPD is being treated, and we have very good indoor patient facilities for large animals, small animals. We have pet spa, horse equine clinics, wildlife clinics. We have very good radiology and imaging center. So also for the instructions of the students, We have instructional livestock farm complex where in addition to the CBF where Gir and Jafrabadi buffaloes are there, we have the Kathiawadi horses, camel, we have with us poultry farm, sheep and goat farm. So also we have a small rabbitary and the instructional farm unit also has Gir animals and Jafrabadi buffaloes for the research and education purpose in the veterinary faculty. The veterinary faculty over a period has grown right from 30 intake capacity to at present 84 per year as per BCI. And we have very good laurels to best of our flank under the umbrella of Junagadh Agriculture University that many of our students have visited foreign countries, secured good markings in the senior research fellowship and junior research fellowship at state and national level. We have a very good student component where students are having good sports complex, hostel facilities. So also we have with us the very good health facilities on campus. The students are achieving many milestones in the COVID virtual that in India, we are the first to conduct online annual examination as per the guideline of Veterinary Council of India. With this brief introduction, I request Honorable Vice Chancellor and President of the function to inaugurate the function by lighting the lamp.
Thank you very much. We are very lucky to have a very distinguished chief guest because Dr. Motilal Madan sir. He renowned for his varied and distinguished work in education, research, and development, and in the science, education, management, administration, which had a meaningful impact on the individuals, institutes, and ecosystems. Now, I would like to request Dr. M. Madan sir to have an address from the chief guest. Madan sir, over to you.
Namaskar, Madan Sahab.
Namaskar.
Sir, we have with us our Honorable Vice Chancellor, Dr. V.P. Chogotia. He is PI of the Institutional Development Project, Director of Research, and he is expertise in plant breeding by his subject or discipline. And he is with us today as the President of the function. I am also flanked by Director Extension Education, Dr. Gajipara, Dean of the Engineering Faculty, Dr. Bhotia, Dean of the Agriculture and Agribusiness Management, Dr. Pont, Nodal Officer ICR, Dr. Mohanat, Student Activity Director, DSW Director, Student Welfare, Dr. Malam, and the Organizing Secretary, Dr. Wada with me. I hope some of the faces may be known to you. I request you to please, after this brief introduction, bless us with your laurels.
Yes, it has been very pleasing, and I will say that it has been thrilling for me to be amongst you for this inaugural function. Often subject which needs more elucidation and more explanations as far as our progress and development in the country is concerned. Honorable Vice Chancellor, Dr. Dean of the Dr. Tong, I see Dr. Kavani around there, Dr. Guman. Pleasure in seeing you around here in this webinar. First of all, I think that it is opportune for me to go back onto the Honorable Vice Chancellor for having occasioned to get this webinar going. And the way I have been with these webinars for the last 3 months, but I see that the deep Prajalan in true sense being held over in here, where I could see that the Deep Shikha and the illuminance is going to spread not only into the room in which you were seated, but it is going to go around to all homes, all desks, all faces, and all rooms where you are being heard or we are being heard at this time. And at that note, Junagadh is a lovely place. The university is doing very well. I had occasion of being there a couple of times and have always enjoyed the progress which the campus has made. And I'm fully appreciative of the effort you are putting in seeing that the education, not only in Saurashtra, but in leading in certain parts as far as the country as a whole is concerned. Friends, today I am personally thankful for having provided me an opportunity to be amongst you, particularly on a subject that with I have been and with which it's always enjoying to talk about. I remember that when we, as a reproduction man, I was introduced to immunology, was when I put in my papers for my graduate school in the United States. And my graduate committee immediately put me to have 3 courses in immunology, and that too in the medical school, because the medical school immunology was considered the best immunology being taught over into the campus. I was really shattered. I said that why after all immunology for me? I have come for reproduction. I have come from reproductive physiology, but why immunology? But my advisor told me that since you are working in the area of immunology, so I could not figure how I was working in the area of immunology. But I realized it soon because I was supposed to be working with antigen and antibody. I was supposed to be working on hormones, produced antibodies to hormones, and through these antibodies, estimate micro quantities of hormones, and then put these antibodies over into the system and ensure how the regulation of the hormones takes place over into the body. I think ever since that time, that immunoreproduction has been always very fascinating to me. Not only because that it reveals many of the secrets of reproductive process, but it also gives answers or attempts to provide answers to several problems which we feel are there existing in the field with our livestock and our livestock owners. As a matter of fact, getting back onto these antigen antibody reactions, what we call as subsequently radioimmunoassays and enzyme immunoassays, which helped me to rather go into the area of investigation of hormones over into the reproductive system. And it was this initial exposure of strong immunology and the working of immunology in the body, antibody antigen reactions, and the defense system in the body, immune defense in the body, that subsequently that we could tackle some of the diseases like anestrus conditions, disease conditions like anestrus and repeat breeding in our cattle. This training, initially I should share with you, was responsible for me, rather helped me in having as many as 12 hormones from a single blood sample here in my own country once I got back using these reproductive technologies. But where we are, that we were firstly at that time, we were talking on a macro level. We were talking, understanding the functioning of an animal. The animal settles to service or it doesn't settle to service. And if it doesn't settle to service, what could be the reasons? Again, the reasons we used to talk often are macro reasons. But since then, from those macro reasons, we have year after year been reducing our insight at the same time broadening our understanding. I said the two things simultaneously because we try to know more and more of a particular thing. The more we know, it means we are concentrating on less and less. Or in fact, that at one point of time, we are supposed to be knowing more or everything and least about nothing. And that is the enigma with which we are today as far as the immunoreproduction is concerned. We have gone a long way in understanding about this immunoreproduction, but at the bottom of it, we say that we know really very little. Because I remember that we shifted from the macro level to understanding of the chromosome. Then from chromosome, we found out, we did the analysis, chromosome analysis, and found out that yes, there were abnormalities which were manifest over into the animal reproduction. But then we found that manifestation of these abnormalities is not a single function of these chromosomes alone, but there is something beyond that. So we went about into the gene constructs. From gene constructs, we went over into the short satellites, that trying to find out that if this is a combination of certain genes which is bringing about. Finally, we went still down over into a situation where we went and said that, yes, it is this gene which is responsible. As a matter of fact, from the time that the bovine genome was announced, we tried to cry panacea, that we have really, and say that this panacea is in fact a eureka, and we have just reached where we wanted. But again, we found that that one step forward was several steps back. We went over to getting over into the DNA. From DNA, we tried to find out that, yes, what are the genes which are expressing. So the gene expression started coming in. The concepts of gene expression came in. The protein synthesis came in. The up and down regulation of protein synthesis, gene expressions came in. And so the explanations which earlier we used to provide to a large extent got changed. And today that we are again at several crossroads, because on one hand that we want to increase the fecundity, we have to increase the number of young ones born from our people, from our livestock. And at the same time, we want that it should be done in an optimal condition so that we are totally increasing the animal production and productivity. So addressing these two things, friends, it's a pleasure that you people have got together there and given us an opportunity. Two things come to my mind at this time when we are at the inaugural function. One is with respect to that seeing the animal as a whole, seeing these animals as part of our production system. And there are no two opinions that in this country today, that it is the livestock that which is the backbone of our economy. Agriculture economy in particular. Why I say agriculture economy in particular? I mean, because over the years, over past decades, agriculture has been having a growth. Total agriculture has been a growth about 2.5 to 3% over the years. sometime less, sometime more. And this, when I say sometime less, I mean that the agricultural growth per se in crops has been even less than zero. It has been minus. But what has been this strength which the agriculture has, which that we were clocking almost all the time around 2, is this net 4% growth in animal science and in livestock, that which propelled us to a total growth in agriculture. And this thing has been realized very much in terms of our growth structure, believing that agriculture is the primary growth engine of our country. That herein, this engine, the molecules which form this engine, I will not say the wheels or the body, I will say the molecules which form are millions and millions of animals. And along with them, millions of landless farmers. So here it is the criticality that on one hand, we are talking of this animal being the pivot around which our ruler, our agricultural economy is dependent. On the same time, we are find that this is a very impoverished entity in the total agricultural structure. The land holdings, as you know, are very poor, and over and above that, about 59% of the livestock is being held by those who do not have a land. And so really, to me, I always address these people not as farmers, but as animal keepers. Because farmers, at least to my concept, are those who have got a piece of land to attend to. But here it is that these have no land, but they survive. They not only survive, but they make sustainability to our agricultural production system. Sustainability comes in terms of the products which come from animals, with the milk, the meat, the eggs, the wool, the offals which come from our slaughterhouses, and the other products which are made from livestock. Now, if we consider them in totality, we know that at present, the amount of GDP contributions which come from milk alone is more than the wheat, maize, and rice put all together. So their contribution is more than these, and therefore the importance comes in of livestock. And the importance of livestock is not in having an animal. The importance of the livestock is that having a productive animal. And to us, production means reproduction. If the animal does not reproduce, there is no production. As a matter of fact, all these production items I was talking about, whether it's an egg, it's meat, or it's milk, they are byproducts. The main product is the young one. So friends, what an excellent thing that you are doing, that you are taking care of this area where we are talking of that how these young ones are born, how optimally they are born. The concept goes to fertility. What is the fertility? How does this fertility get controlled? And what are the means by which we can increase fertility? At this time, it is my pleasure to have been associated in this program. And I'm sure that we'll have an opportunity to be sharing my thoughts in greater detail subsequently. Thank you very much.
Thank you very much, Professor Dr. Ahmed Malan, sir. Giving the very brief introductions regarding the title of this vocational training. Now we have with our guest of honor, Dr. A.K. Kawani, sir. We would like to have some words from Dr. A.K. Kawani, sir. It's over to you, Dr. Kawani, sir.
Hello, Kawani.
Everybody.
Kawani sir, namaskar.
Namaskar, sir. Respected President of the Function and Vice Chancellor of the Junagadh Agriculture University, Dr. Chotia sir, Dr. Madan sir, chief guest of the functions, my friend Dr. Gajipara, the Director of Extension in Junagadh Agricultural University, Dr. Khun, Dr. Malam, the Dean of the Faculty, Dr. Tank, Organizing Secretary, Dr. Wala, Co-Organizing Secretary, Dr. Sharma, my colleagues, Dr. Dhami, Dr. Panchal, Dr. Suthar, Dr. Goham, and the dear students. We know that India ranks first among the world milk production nation since 1998. As the largest bovine population in the world, milk production in India during period of 1950-51 to 2019-20 has increased from 17 million tons to 187 million tons. As compared to 176 million tons during 2019, current growth rate is 6.5%. Milk production per cow has steadily increased through the combined improvement of management, nutrition, and genetic selection action. Total bovine female population in India is 136 million, comprising of 81.35 and 54.98 million adult cows and buffaloes respectively, as per the 20th livestock census. In Gujarat, total bovine female population is 10.16 million, comprising of 4.5 million and 5.67 million adult cow and buffaloes respectively. With the increased number of high-producing cow and buffaloes in farm, maintaining reproductive efficiency at an optimum level has become a challenge. Thus, the high-producing female have a higher incidence of infertility or subfertility. Other stress factors affect the reproductive efficiency negatively, besides the milk production stress. Among the animal inseminated, rate of fertilizations has been estimated to be greatly high, up to more than 90%. But the overall rate of detectable pregnancy had crosses to 40 to 50% in high producing dairy animals. This fact indicates loss of embryo pregnancies at an early stage up to more than 50% due to multiple causes of either male or female infertility. In many cases of infertility, still it is not always possible to determine the cause of reducing production or infertility. Poor reproductive performance of the animal leads to economic losses due to reduced production and additional cost of management. Impaired reproductive functions are reflected in the form of anestrus, prolonged dry period, delayed maturity, decreased calving interval, and the lifetime productivity of the animal. About 30% of cattle and buffaloes are culled for the infertility issue, losing the valuable genetic resources. Those incur direct losses to the farmers over the increased cost of management. Accordingly, the economic loss can be in the form of loss of thousands of liters of milk, calf crop, replacement stock, culling, and veterinary inputs. A study conducted at LRS NDRI Karnal in 2015 recorded a loss per day to the farm due to an increased open period by 1 day in Sahiwal and Karamfish cows to be rupees 281 and rupees 368, having an average milk production of 7.3 and 10.5 liters respectively. The causes of infertility are many and complex. Apart from the known causes of fertilization failure, naturally occurring cross-reacting antibodies have been said to be responsible for infertility in human and animals. Anti-sperm antibodies developed against the spermatozoa have have been said to be detrimental to the sperm fertility, causing subfertility, infertility, and repeat breeding in animals. Several hypotheses are given to explain the development of anti-sperm antibodies. Multiple evidence through research reports are available in human infertility, but little is known about the immunological infertility in the animals. This fact asks for, however, through investigation and discussion on the immunological aspect of infertility in domestic animals to optimize the reproductive efficiency and to prevent the economic losses to the farmers. The participant students of this innovative study program are suggested to have a critical look into the various facts of the infertility to have better understanding of the etiological involved as to resolve the infertility, to optimize the reproduction for increasing the milk production, to prevent the erosion of valuable genetic material, and ultimately to boost up the farmers' economy and the national GDP too. I would like to congratulate the organizers for the selecting the topic of this training, which is need of the hour and justifiable. Thank you all.
Thank you very much. Dr. Ravani sir has described the different causes which are occurred in the male and female infertility, how the immunology is acting role in that. Thank you very much, sir. And now we are very lucky to enough that we have with the Honorable Vice Chancellor of Junagadh Education University, Junagadh. Dr. V.P. Tovatia sir is available with us. So I would like to invite her to give the blessing address for this vocational training.
Namaskar. On the occasion of the inaugural session of vocational training on immunological advances in male and female fertility, today's chief guest, Honorable Dr. Madan sir, guest of honor, Dr. Ravani sir, Dr. Dhami sir, our Director of Extension, Dr. Gajipura, Dean Faculty of Agriculture Engineering, Dr. Gotia sir, Dean PGI ABM and Principal, College of Agriculture, Dr. Khont sir, DSW, Dr. Malam, Dean Faculty of Veterinary Science, Dr. Tang sir, OPI, Dr. Monat, Organizing Secretary, Dr. Wada, Organizing Secretary, Dr. Sharma, other participants and friends. As I am not expert of this field, so I have no right to spoil your valuable time. Even though I would like to suggest one thing, that we have a CRP project on artificial insemination in Gir cow and Jaffrabadi buffalo. As seen after the report said that the yield increment in Gir cow is approximately 37%, and in Jaffrabadi buffalo it is approximately 20%. But I do believe that if the male and female fertility is increased, because as a layman I know only that the fertility ratio after AI is near about 80% in Gir breed and 70 to 75% in Jaffrabadi buffalo. If this ratio will increase, then definitely we are able to increase the productivity of the milk. So I humbly request all the learned members and all the experts to done something and find out the way to increase the productivity by the different tools. So without losing your time, I wish to grant success of this occasion, this training, and make it fruitful with your all positive efforts. Thank you.
Thank you very much, sir. Honorable Vice Chancellor Dr. Vipin Jawatiya sir said, if sir you are not expert, okay, but your vision, your focus and attitude to develop institute is very important for us. Thank you very much, sir. Now, before the vote of thanks, I would like to say that after the completion of this inaugural ceremony, we have with the two very important and the eminent scientists are with us to discuss the two lectures. So keep it with us. Now I would like to invite Dr. A.D. Wala, Organizing Secretary of this whole vocational training, to have a vote of thanks. Please, sir.
I propose vote of thanks as an organizing secretary of this 5-day online vocational training entitled Immunological Advancement in Male and Female Fertility. At the end of this inaugural session, first and foremost, I would like to share my sincere regards to our Honorable Vice Chancellor, Dr. V.P. Jawatiya sir, for providing us all the support to make this training fruitful for our beloved students. Thank you very much. I would like to thank Dr. H.N. Gajipura sir, Director of Extension Education, JE Junagadh, for remaining present in this function. Thank you. I would like to thank our Principal and Dean, Dr. P.H. Tank sir, who always stand among us for support and necessary guidance as and when required. Thank you very much. I also thanks to Dr. N.K. Gotia sir and Dr. K. Khut sir to remain present in this inaugural function. I also thanks Dr. V.R. Malam sir to remain present in this inaugural functions. I also thankful to Dr. P. Mohanad, ADR and Nodal Officer, IDP, who always ready to face the problems and provide the fruitful solution. Thank you very much. I also thankful to Dr. Madan sir and Dr. Kawani sir to remain present and give valuable guidance for these trainings. I would like to thank all the HOD, faculty, non-teaching staff of the Veterinary College for unconditional supports. I again sincerely thankful to convener, co-convener and member of all the committee who devoted day and night to make the training grand success. I also thankful to IT director who provides the internet and other facilities. I also thankful to all the person involved directly and indirectly to make this training successful. Till 1100 participants in this training joined. I once again thankful to one and all. Thank you very much.
Thank you, Dr. K.D. Wala sir. Before we end this ceremony, we would like to release our CD and compendium of this training. This compendium and CD contain all the lectures which are to be given by these 7 speakers, and the training modules of the practicals are to be contained in this compendium. So, we are going to release a CD and compendium of this training. I would like to invite Dr. V.P. Jawatiya sir to release the CD and compendium of this training. Please, sir. Thank you very much to all the dignitaries. The soft copy of this cooperative will be sent to all the participants in their mail. Now, the end of this inaugural ceremony, we would like to raise at the seat and we have a national anthem to end this inaugural ceremony.
विनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाए तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे भारत माता की जय
Thank you very much. In very few minutes, we are going to start our first technical session. We will start our first technical session at 10:30 a.m. So all the speakers and the participants are requested to remain present at 10:30 a.m.
જે બીજું હોય એ તો વાત કરી
What happened? Is all okay?
હવે ચાલુ થશે.
I hope things are going well.
આપણા કેમેરા મ્યુટ છે ને સાહેબ, એટલે આપણો અવાજ ના જાય. નહીં તો જાય.
અવાજ જાય નહીં.
નહીંતર જાય. ફેર પડી જાય. Resolution ફેર પડી જાય.
સાઈઝ નાની હોય ને એટલે સારું.
હું ચાલુ રાખું છું. મદન સાહેબ નું હલો, આપણો અવાજ પકડાય છે.
તમારા
Again, very good morning to everybody, respected speakers, and all the participants from the all over India. So, we are going to start our first technical session, and which is to be held by Professor M.L. Madan sir. I would like to narrate a brief resume of Professor Dr. M.L. Madan sir. Dr. Madan sir is a his significant achievement, the outstanding scientific leadership in science and technology contributed significantly and impacted national development. He began his professional career from Haryana after completing advanced study in the United States, and he always been focused on the betterment of the people. Dr. Madan sir is currently Chairman Research Advisory Committee of NDRI Karnal, Chairman QRT, CSWRI, Avikanagar, Member Outcome Review Committee Scheme of ICR. He is also a member of Planning Board, Kamdhani University, Gujarat, and he is also a patron, the SAPI of the Society Animal Physiologist of India. He was a former Vice Chancellor of Pandit Dindayal Upadhyay University of Veterinary Science, Mathura, and the Vice Chancellor of the Punjab Rao Deshmukh Agriculture University, Maharashtra. He also served as a Deputy Director General Animal Science, ICR. He was Joint Director Research and Project Director Biotech, NDRI Karnal. He was also a chairman and president in different national societies and committees like Haryana Farmers Commission, Society of Animal Physiologists of India, Research Advisory Council IVRI, National Task Force in Animal Biotechnology, and also Education Committee VCI. He was the expert member in animal science and fisheries in the Parliamentary Committee to evaluate the research work of ICR Institute, DAR in New Delhi. He was also a FAO consultant for the buffalo development in Iran. He was Honorable Advisor Livestock Development, Government of Sikkim, and also he was a member in many expert committee of national and international projects on cattle and buffalo development. His primary education, he did his BBSc age. He was a gold medalist from the Punjab College of Veterinary Science and Animal Husbandry, Punjab University in 1959. He was MSc gold medalist, NDRI Karnal in 1965. He did his PhD from University of Columbia, Missouri, USA in 1971. And he also got the honoris causa, the Director of Science, DSc, from the Chandrasekhar Azad University of Agriculture and Technology, Kanpur, and the University of Gulf, Toronto, Ornia to Canada. With these brief introductions, I would like to invite Dr. Madan sir to give the lecture on immunological issues and advancement in the fertility. Over to you, Madan sir, please.
I'm on the screen now. Is it audible? Is the sound audible? What's the problem? Is it on the screen?
Yeah, yeah.
Yes, yes.
And is the sound audible?
Yeah, audible and the PPTs are on the screen. Yeah.
Okay, well, that's wonderful.
And
I think that's a lovely time to begin then. At the outset, that thanks for the introduction which you provided. What I believe has come in the way of introduction should be prefixed with this word reproduction. Because all what I have been all these years doing is working in the area of animal physiology and touching the various aspects of physiological functioning to augment productivity and production. Friends, today I'm very amused by the title which has been given to me in terms of the immunological issues and advancements. When I was looking into it, I found that there is a large number of speakers who have very specific things to contribute. I thought that why not I touch issues which probably will encompass all of them and they will amplify on them. And I have opportunity to express myself on some of those things which are very vital in terms of fertility. To begin with, I would like to share this picture with you all. This is the most fascinating thing I have ever seen in the world. Having had opportunity to go around the world, seen almost every wonder of it, and having been associated with education and read about several subjects, I am yet to see anything which can beat this little structure, which I believe you must have understood by now is embryo. Here it's an embryo with the granulosa cells around it. The life starts there and the life ends there. The toughest thing ever produced in the world, where no entry can get, is the structure which is having this covering on the outside called the zona pellucida. And when we are talking of fertility, we are addressing, in fact, ourselves to this little structure, which as you look at it, you may not be able to see anything. But friends, when I look at it, I see each one of you to have started right from there. I see all your abilities and affiliations getting created out of this embryo. And I see this as a short emblem, as a short history of whole evolution in the world. If you see in true sense that what is there in this embryo, when we see at one stage, that is when this ovum is produced and gets out of the ovary, and till the time the young one is produced, the type of metamorphosis which takes place. How does metamorphosis occur? From a single cell, you have a different structure of eye being formed, liver being formed, testes being formed. And the greatest of that, as a physiologist, I say, there is no better auto-regulating system in the world as of today than a kidney of an individual. And one wonders that how does this embryo get itself into make all these structures? And as part of that, the unique thing is that this oocyte cannot get into an embryo until it has a structure coming over into it, which we call the process of fertilization. Though at present we have technologies with us in which we do not need that process of fertilization per se. Today we can produce a young one from the air cell of an individual. We can produce a clone from the cell in the semen, from a cell in the semen, non-sperm cell, epithelial cell in the semen, we can produce a young one. But the biologically, I'm addressing myself to this unique structure. And when we refer to fertility, as in fertility refers again to the capacity of this ovum meeting the egg and the union taking place. I have, therefore, to say that this union of egg and the sperm, which lays the foundation for life, and that life is connected with which today the livestock. And this livestock is a critical thing by which sustainability, nutritional abilities, the nursing mothers, the newborn child are all able to sustain themselves in this country. And this, what I call the state of this livestock, is different in terms of reproduction. Because when we talk of reproduction, we always say that, yes, we refer to the production. But the manner which it is in India, that where the resources at the command of an animal to reproduce, is completely different. And the system mode is completely different. So as a matter of fact, when we are talking of when issue of fertility, we have to address the issue of the system in which our animals are, the production system. And in that production system, we are talking of an integrated production system. I wonder whether any one of you is gone about in your thinking about 70,000, 80,000 years back. And that time when the Homo sapien came up, he was just like any other animal, scared of other animals living in caves. And over years, in the process of evolution, a thing has occurred when the Homo sapiens came in, a concept of gender came in, that it became a male and a female. And this male and a female is as a result of evolutionary economy. The male is the specialized function of only producing is an economic evolution. And the female is having the bundle of cells and is also having the capacity from those bundle of cells to nurture the young one in which different ways, whether it is oviparous and ovoviviparous or viviparous, but capacity to nurture the young one. And the uniqueness of this evolution is that the female conserves itself, the process of conserving by having only one ovulation over a period of time. And unique about animals is that it invites attention at the time of ovulation so that the coupling can take place, so that the mating can take place. So therefore, the concepts of fertility come through this evolutionary process in which that the female prepares itself and calls out for mating for the young one to be produced or the process of fertilization to take place. And this happens because the female has the capacity of receptivity to mating. And it is this which sets the process of whole reproduction on. And then comes the other things. It doesn't look to be that complicated when you look at a larger view of it. That we have the male and the female and the young one is born. But when we look a little more indirectly, that we know that this process is through production of gametes. And this gamete production is a process in itself. It's a very circumcised and very regulated process of gamete production, in which it is not one system, but there are several systems in the body which are involved to the production of a gamete. And it is not only that production of gamete, but delivery of gametes that which completes the process of reproduction. And that is when the pregnancy sets in. And it is followed through the act of fertilization. When the gametes meet, the zygote is formed, we say this is fertilization, which leads to the gestation. And in that all process, there is one thing which is supervening, and that is the science of immunology. And that's how that I want to impress that how this immunology is so important in the area of reproduction. And therefore, the concept of fertility is born. That when we talk of fertility, it may be a small word, but it is having all these connotations with which I have addressed myself. Now, in terms of immune structuring, that I'm talking of two types of responses within an animal when we talk of immunology. The humoral immunity, which involves the manufacture and secretion of immunoglobulins from B cells, and cellular immunity, which involves various subpopulations produced from T cells. So in fact, that when we are talking of immunology, what we are referring to is the immune responses which is directed against an substance called antigen and characterized by great specificity. That's how, and in all mammals, we have what we refer as the basic of immunoreproduction, and the basis is this histocompatibility gene complex. The codes which are on the cell surfaces, proteins and cells and tissues, and these proteins are markers for self and are unique to the individual. They are usually glycoproteins and are involved in recognition and immune response to foreign antigens. Any substance which comes outside or which is external to a moiety, we refer to as the antigen. And therefore, this antigen appears all over the body. They are in the hypothalamic cells, they are in the pituitary, they are in the gonads, they are in the placenta, they are in the fetus. The concept when we talk of immunoreproduction, immediately it goes towards the reproductive tract and it goes to the reproductive cells, the gametes. But no, when we are talking of immunology of reproduction, we are talking of the hypothalamus, pituitary, the gonads, the placenta, which is the temporal gland, and we are talking that there can be specific antigens which could alter the expression of these all function of expression or function of all these things. And what alters that? As these antibodies that which are in there, and these antibodies will be bringing about that against these, whether they are the hormones, they are the hormones from the pituitary, from the gonads, the steroid program, or those which are associated with the process of fertilization, gestation, parturition, and the birth of the young one. Antibodies against antigens of reproductive tissue and hormones can be used to quantitate and qualitate, destroy, neutralize, or enhance the specific reproductive events which are associated with normal fertility or infertility. And that is what today the jargon of immunology is. That it's not only the concept of antigen and antibody, but it is that we can quantitate and qualitate. We can use these principles for quantification and even for destroying, neutralizing, or enhancing their reproductive events. And it is also problems in inducing antibodies to reproductive antigens, the heterogeneity of immune response by individuals, whether it's an inbred or an outbred population. And as far as we talk the nature of this immunologic response, there is no strong or weak antigen. Very strong antigens can become very weak, or very weak antigens can be very strong, only in high or low responding on the part of the animals. A key to the development of antibodies is to turn genetically to low responding individuals to high responders, incorporating the antigen with an adjuvant to provide the phenotypic correction. Response influenced by both exogenous factors such as stress, nutrition, and endogenous factors such as circulating concentration of hormones. They can change the response to any antigen. For example, best example of this is our testis barrier for spermatogenesis. After having, as I mentioned earlier in the morning, after having had my first introduction to the immunology through my research, the first introduction to operative immunology was in my laboratory here, when I found the then director of the National Institute of Immunology coming loaded with a syringe of antigen and then attempting to produce aspermatogenesis as a means of fertility control. One of my graduate students, Dr. Suri, who subsequently headed that institution, was involved in this work, which we started by simply injecting into the testes and then gradually seeing that how they became azoospermic over a period of time. And this type of response, because we were breaking the barrier, we were injecting it right into the testis itself. But under normal circumstances, these antigenic substances do not enter over in there. There is a concept of what we call as isoimmunization of female to sperm. The female reproductive tract is capable of generating an immune response to ejaculated sperm that can result in infertility. In bovine, such type of immunological infertility is rare because that antigenic response to semen is once in 21 days. There is no continuous load of semen in the female reproductive tract. And that too, when the load of semen comes over in their bovines, at the time when it receives the mating or it is inseminated, at that time it has just come out the heavy estrogen feed, heavy estrogen load. Neutrophils that normally phagocyte sperm cells are elicited by the non-antigenic stimulus that destroys excess sperms without providing antigenic stimuli. And also that the phagocytosis in the uterine secretions flushes sperms from the cow's tract without stimulating the new flow nodes. And that's how that we see that in bovine that we don't have much of this isoimmunization. But this happens to be one of the major problems for human reproduction. Today, when we are talking of artificial reproductive technologies, and we are talking of IVF in human beings, that one of the major issues which comes out is this immunological incompatibility, which develops out of the semen and the reproductive tract of the female. If we talk in terms of fertility as such, you'll be surprised that normally about 67% of all those fertilizations which occur, that means a zygote is formed, does not result in pregnancy. And when we want to if we want to analyze that what is this infertility due to, it finds that about 30% of infertility is due to female. About 30% of infertility is due to male. There are about 30% which are unexplained, and about 10% in which the combination of all the three are there. And therefore, that whenever we want to talk of immunoreproduction, what is the agenda which we want to be talking about? We have to talk of linking the immune and reproductive systems. We are talking of immunogenetics as the major immunocompatibility complex. We have to interpret gamete interaction with their stromal cells, gametes, and antigens. Gamete gene expressions and regulation. We have to express the mechanisms by which early embryonic developments take place. And the criticality is the maternal fetus interactions. Most of the attention these days in terms of fertility, whether it is in animals or it is in human beings, is towards this aspect, that early embryonic development and maternal fetal interactions and pregnancy maintenance. This is what we call as the crosstalk, which goes on between the maternal membranes, fetal membrane, uteral membranes, and the fetal membranes of the developing embryo. We also are talking of the modulations which are taking place in other hormones by the immunological modulations, which refers to immunogenesis or lactogenesis. We are talking of a battery of proteins, which refer to as pregnancy proteins. And our attempt these days is to identify one of these proteins as the pregnancy factor and assess fertility through sensors. If we have got these proteins, about which I will be sharing some information with you, if we are able to isolate these proteins, then we have a sensor. We have just to go on to and find out about the pregnancy. We have got about now this nanotechnologies by which we can deliver the sperms, and we can also talk of nanotechnology using in immunomodulation. And finally, that we have also to be talking about what we refer today as the artificial intelligence, and artificial intelligence, how we can overall and how we can control the natural phenomena which are occurring in the process of sperm modulations, ovomodulations, and fertility and infertility situations. I have already explained about the antibodies to antigen as far as the animals is concerned. But there is a very great factor that whenever we take the semen into the female reproductive tract for insemination, we are simultaneously challenging the female reproductive tract, not only with the AI for semen of the bull, but along with goes that the egg yolk diluent. And these antibodies to egg yolk in 48% of the cows inseminated, or 3 times, and 58 of those inseminated more than 4 times. These, whether you use the egg yolk or you use the milk diluents, they produce antibodies. Vaginal antibodies to egg yolk are about 70 to 80% respectively in those which are primiparous and they are multiparous animals. Infertility with freeze-dried semen protein stability in aqueous medium is depending in part on that what is the content in there, and the removal of the water and the way it is watered, because it leads into the restructuring of the protein. And restructuring of the protein can also become antigenic in functioning. Although as of today, that we have got a number of methods by which that we try to approach the problem of fertility, but we know that 20 to 40% of the bulls on the male side are having reduced fertility. There may be no single diagnostic test as of today, but we know that we have got what we call as a battery of tests used through a called the CASA system, where the evaluation system is being done, by which we can find out that what is the gross picture of the semen. The recent developments in the prediction of bull fertility based on assessments at molecular, cellular, and whole animal level are also in progress. When I talk of molecular, we are talking of the gene action. When we are talking of cellular, we are talking of the cells itself, and whole animal is that we are talking at the gene level where these are being studied. High fertility spermatozoa differ from low fertility spermatozoa in some ways. And I have just listed 4 major ways. Number 1, compared to the sperm of high fertility bulls have a higher expression of proteins which are involved in energy metabolism, cell communication, supermetagenesis, and cell motility. Therefore, the bovine spermatozoa proteome is now very critical in evaluating the fertility. Over on, we used to see that we used to mate a male with about 100 females or 1,000 females, and then find out that how many of them have become pregnant and give a fertility rate. Then we became a little more sophisticated. We said that, all right, let us test in the laboratory that this semen, how much of zona-free eggs it's able to fertilize. So we talked about that fertility rate in terms of fertilizing capacity. But now, with the modern technology, we are now able to do what we call the metabolics. We are talking of omics and proteomics. And through these measures, the bull fertility is being measured. The surface cell antigens, that is THY and OXA, are related to the follicular growth and atresia. In rat ovaries, the Thy1 and X2 are non-growing, are also antigens for non-growing follicles. But in bovine, the Graafian follicle rupture during the metaestrus is approximately 24 hours after LH surge. And it is hypothesized that this rupture is due to the inflammatory reaction. And in this inflammatory reaction, we have got gene expressions. And lately, that there are 3 genes which have been associated and related to the follicular growth and ovulation. Now, I already said about this, but there is what we call as autoimmune aspermatic orchitis. The environment of blood test is barrier. Sperm-specific molecules foreign to the male can be synthesized and inserted in the plasma membranes of primary spermatocytes, secondary spermatocytes, or spermatids. The exposure of these sperm-specific antigens to the immune system can cause immune response that results in what we call as autoimmune aspermatic orchitis. There are other areas. For example, we are talking now of specialized bodies to specific parts of the protein. That's we call as the monoclonal antibodies, and a bovine sperm epitope has also been used to interact as far as the epidermal antigens are concerned with the female and male reproductive system. In the human story, that we have got antisperm antibodies which are invariably present. But I'm not saying much about that because it is said, it is strongly believed that in human and other species, at least some antibodies may bind to sperm antigen causing infertility. But such a situation is only, I would say, rare as of today. These antisperm antigens, as well as they're circulating in semen after sperm production, sperm transport, implantation, early embryonic development, genetic interaction, and fetal development. People have reported seminal essays that in
Sir, respected sir, your mic went mute. Madan sir, good morning, sir. Sir, your mic is mute, sir. Madan sir, kindly unmute. Thank you so much, sir. Thank you.
when did it happen? I hope you are able to listen all right.
Yeah, we are just 2-3 minutes it happened. No problem,
Oh.
sir. Thank you so much.
Oh,
It's okay, sir. Thank you.
all right. We have these anti-sperm antibodies, as I was speaking to these, and we have data from cows which show that there is related to their reproductive health. Those which are having an high values, significantly high values, and are positive, or those which do not have an visible infertility, but which are having a high ASA or having infertility in bovines. For example, I'm quoting the analysis of predicting bull fertility is that a study in which 537 cows were examined by ELISA test for anti-sperm antibodies. In 420 pluripellis cows, the incidence of positive findings were significantly higher than in about 117 primiparous cows. Significantly lower penetration and velocity of bull spermatozoa through cervical mucus was recorded in heifers serologically positive to ASA after experimental immunization. And there is the actual physical data which shows the difference, and in the different laboratories between the different anti-sperm antibodies. I don't think I need to go into details on this, but suffice to say, the graph which I showed you, that the difference which comes over into this. And also the difference, the extent of anti-sperm antibody IG binding along sperm tail is finding, and then the percentage motility, the number of sera, the relation between the anti-sperm antibody IG binding and motility. This gives us a picture that one of the reasons in terms of fertility in bovines. We are also talking of this molecular reproduction and development, in which that we are talking of this basement membrane, which serves as an barrier to the incoming proteins. So the factors which are affecting the blood testis barrier. Various people have worked over this, and a host of these cells have been identified over in there in this basement barrier till the time that spermatogenesis is occurring. So we are having what we say the basal production cell production over in there, and then we are having the supermatids and spermatozoa formation over in there, and how these basal membranes, if they are interfered with, will let the protein antigen molecules to go up over into the supermatids, and it is in supermatid stage that beyond that stage that you find that immunological reactions takes place with the sperms. Also to be understood here is a very important new concept which has come out over into the follicular development. Before you is a follicle, in which you see that the follicle is in the follicular fluid, and there are these cumulus cells which are around these. Now these cumulus cells are now accountable to a large extent for the process of fertility. Subsequently, because it is these cumulus cells, the protein actions which are from this, which is causing basically the process of ovulation. The proteins which are synthesized, the lytic proteins which are synthesized over this as a means of gene expression, which cause the process of ovulation. And along with this, that we are seeing certain types of these proteomes which are coming from the blood cell and from the blood circulation, and some which are de novo synthesis by the thicker internal cells in there, that those are supposed to be the ones which in put a barrier towards the fertilization of the ovum subsequently in the process of fertilization. And through the microarray, about 47, about 117 such proteins from the Graafian follicle, especially at the type of ovulation, have been isolated. And out of these there are about 7 of them which have been found to be critical. Similarly, that non-inclusion of chromosomes can overestimate the effect of autosomes, and not only because of additive effect of X chromosome itself, but also because of the epistatic effect of autosomal genes. I'm referring because X chromosome is the usually the largest genome-wide association, and it has not been very much included over in the process. But genes in this X chromosome are involved in reproduction. Reproductive traits about sacrotal circumference, early pregnancy, age at first calving, in Nillor herd genotype data from 3,263 animals have been shown that they are having a particular type of genotype. And for P16, the effect was increased to almost 20%. The same pattern when we are finding that increase in more than 10% for important twice and important windows, that where we see that we have got the infertility situation coming over into the males. The male factor infertility presents a much more waxing. That I was talking to the female. Now about the male, it's much more, because many studies have found that abnormal sperm parameters using mass spectrophotomy, the study of sperms and more specifically sperm proteins. The aim of this study, which was done, was to review the literature and also to do this proteomic techniques and verify the proteomic techniques and sperm dysfunction. And a large number of proteomes have been identified that through this spectrophotometer of the fluid which is in the female reproductive tract after the semen has been there in the female reproductive tract over periods of time. High throughput proteomic techniques provide new insights in the potential communication between sperm and the seminal fluid on one hand, and then the sperm seminal fluid and the fluids in the female reproductive tract on the other hand. And we have got now biomarkers, potential biomarkers for clinical significance, providing insights into the molecular mechanism that regulate the generation, maturation, and transit of spermatozoa. Similarly, that we have got mechanism responsible for interaction among the essential systems of organism critical for understanding the physiopathology, severely poorly characterized diseases resulting from abnormal interactions. For example, mastitis is one which has got a great immunological link besides several of the diseases. But we're referring today that the immune system, the gastrointestinal system, the reproductive system account for adequate transport of nutrients to the pregnant uterus that is required for growth, development, and survival of the conceptus during pregnancy. So it is here that our communications come in. Summarizing to the long story, The embryo implantation is a unique biological phenomenon and requires a receptive endometrium, a normal and functional embryo, and most importantly, a synchronized dialogue between the maternal and embryo tissues. All attention onto this dialogue today in terms of understanding that what is this happening. As you must be knowing, that the implantation demonstrates an immunological paradox and involves critical changes in maternal immune system, which begins right from a successful fertilization. During the critical period of implantation, understanding the changes occurring in immune cells can not only give an accurate idea of successful pregnancy, but also help in concrete pregnancy diagnosis. The first signal for maternal recognition of pregnancy is interferon tau, which is secreted solely by the tophoectodermal cells of the developing embryo during the process of pre-implantation period. And therefore, when we have to, the main function of interferon tau is to silence the expression of estrogen receptor, that's the oxytocin receptor, to prevent the production of prostaglandin F2 alpha and hence prevent luteolysis. I think it was in late 70s for the first time when the concept of prostaglandins came and the effect prostaglandin was identified as the typical molecule at that time involved in this process of pregnancy maintenance. And subsequently, now prostaglandins have been identified in all tissues, of course, having different functions. But to this date, this prostaglandin F2 alpha is considered to be the critical. And therefore, in infertility situations where we are talking of early abortions, that we still have to focus. Prostaglandin F2 alpha becomes pretty critical when we are talking of utilizing embryo technologies, when we are talking of cloning technologies, and we are talking of preparing the uterus for the reception of the fertilized embryo in the process of embryo transfer. When a portion of interferon tau enters the general circulation, it directly upregulates the expression of interferon-stimulated genes in peripheral blood cells, which can be easily detected. We also say the cellular responses to interferon tau results from their contact with the cell receptors, leading to the expression of many cellular genes. A successful fertilization alters expression of several interferon-stimulated genes. We have listed here ubiquitin-like modifier, ISG15, or ubiquitin cross-reacting protein, usually Ks9, myxovirus resistance genes, MX1 and MX2. These are upregulated in peripheral blood leukocytes, BPL, during the early pregnancy, that is J16, 18, and 20 post-breeding. Myxovirus resistance gene expressions is documented in uterine epithelium, strongly myometrium, and in peripheral blood monomolecular cells of sheep and cattle. The gravid uterus of gilt and mare also indicates upregulation of myxovaccine resistance genes. During early pregnancy, MX2 is strongly upregulated in BPL compared with MX1, with the former showing upregulation of 5-fold by day 20 post-immunization in pregnant cattle. Now, this is where that everybody's attention towards in reproductive immunology is with respect to the pregnancy. And that is the area of what we discuss as the area of, if I put my pointer to it, the implantation. We are concerned with facilitating this implantation, and in the human reproduction, we are concerned with inhibition. And that is where we are talking of genes, upregulation and downregulation, and uncontrolled inflammation and controlled inflammation. In uncontrolled reblation, we will have inhibition, and in controlled inflammation, we have facilitation. We will have a feedback system operating where the negative and positive feedback will bring about the changes in the concentration of interferon and therefore subsequently bring about the receptivity onto the membranes of the uterus. And subsequently, we can see whether it's a successful or it's an unsuccessful gestation. There is excellent data which has recently come out in this area. Interferon stimulation of gene 15 in non-pregnant versus pregnant cattle. And since this is something which is hot from the oven, I thought that this might provide an insight and a lead to people to think into this, that how we are finding that expression of interferon is stimulated by gene 15 in non-pregnant versus pregnant cattle. That we have the non-pregnant and the pregnant cattle, and this is post-AI. And this is a situation which is post-AI. Why I'm putting in this thing, that it is opening one of the most exciting windows over in the process of fertility and immunoreproduction. And that is that if we are able to pick up this molecule, if we are able to get hold of this day 18 or day 21, this protein, which you see in here, I'm sorry, I'm trying to show it by finger, that my pointer has got lost. Okay, in here, we are taking this, it peaks over by day 18 over in here. And in here, I got days post-insemination. And if we can see that day 18, and we are able to pick up, and it will be an what we call as a sensor, pregnancy through sensor, cancer section. And here is also then several cytokines which are also been identified for non-pregnant and pregnant cattle in the peri-implantation stage. These are also being investigated these days immunologically as the proteins and the proteomics. Today, the field is now, immunology is completely different than from where I started probably. I remember that when I started, it was an octronic method in which that we used to take a petri dish and we will put it to agar and put the antigen and the antibody in it and test the cross-reactivity in them. That's how I started. That's how I work at the antibodies for hormones. That's how I have to see that whether there is interaction which is taking place on the hormones and antibodies. But today, the world has completely changed. And where are we today? We are today in the direction of humoral anti-sperm antibodies. We are talking of immune-mediated infertility. We are talking of anti-sperm antibodies of the gametes, sperm in males, sperm in females, diluents in males and females, reaction to spermatozoa involving embryos, immunity to reproduction, and immunity now to find out what with respect to the sex temperatures. I hope you are already knowing that where we were talking at one stage of 10 million actively motile spermatozoa, rather 20 million to begin with. Then we went to 10 million. Today in sex women, we are going 1 to 2 million actively motile spermatozoa. And now you can see the degree of sophistication to selection processes of the males which we have gone to, that we are able to take only about 1 million cells in the ejaculate which have been sexed and then cause a pregnancy and get a pregnancy of about 60% using the sexed semen and with an assurance of about 98 to 99% as far as the sexed semen is concerned. I want to share also in here that if you take the North America today and you want to go over the genetics of all the bulls which are there or the female, you will be surprised that you all boil down to the Y chromosome of 2 bulls of about 10 million highest producing females in North America. What I say that on one hand, that we are in the race for increasing productivity by using this high genetic potential semen, at the same time that we are also to find out the measures by which we'll be able to see the pregnancy. But we have to find also that what about the fertility with respect to these high producing males? Often a concept has been developed that high producing females, high producing animals have got poor fertility. This is no longer true. This has been done away with, this thought. Because earlier, our data used to suggest, unfortunately, we don't have data in this country, but in the countries where there is enough of data of 10 years, 12 years, 15 years, that it used to say that, yes, high productivity animals, low production. But now when they corrected that, for example, in Sweden, in Netherlands, in America, in Canada, when they corrected and stopped the process of that selection from taking only the milk production, they increased 4 other parameters in that. That was the birth weight of the calf, there was the age of maturity of the calf, the calving interval, and the length of the lactation. When they increased these reproductive parameters in these, they found out that vitiation, which was earlier taking with the milk, is now done away with. So in there, the attempts are now over to see that what is the fertility structuring which is taking place. And in the end, I would like to introduce you to the emerging technologies, because where we are, as far as we are concerned, in an era of artificial intelligence with respect to reproductive immunology and reproductive biology. We are talking of omics today, metabolics, omics, RNA omics, genomics, and RNase sequencing. To a point, yes, we say that this will give us a picture, but another overriding thing has come over in this, and that is what we talk as epigenetics. We say that even if we have got the gene may not be able to express. Even if we have now found out that there is an expression that this animal has got this capacity to produce this much, but it is not producing because there is an epigenetic influence. And that is where we now talk of big data. So that old thing is over, and now big data has taken out. That is analysis system, extraction of information of sets up and those which are in complex of nature, and from which that we can do extrapolations. We have got also the imaging analysis, and training for image analysis for software, and we have development of analysis for studying the rate of change or rate of development, which we call as morphokinetics. The morphokinetics studies which are in progress with respect to the embryo in, for example, laboratories in Canada, which I had a pleasure to be visiting and seeing for myself, that it is interesting that we can take a single embryo and we can test the health of a single embryo over a period of time. And we can measure through metabolic studies that how this embryo is going over a period of time, and then transfer it to ensure that, yes, this will be a successful fertility. And this is especially done in reproduction, though this is an animal reproduction laboratory, but a lot of its work is being done with human reproduction. As a matter of fact, this laboratory I visited is one of the unique laboratories which is called the Embryo Health Laboratory. Friends, so we are in an era where the skies are the limits. I don't want to take you to the skies to the limits, but I want definitely to bind you to our own livestock, to the situation which are on ground, to our own artificial insemination programs, our own systems by which we can ensure that the female reproductive tract is receptive enough, the semen is good enough, the semen is having the right fertility. I was rather not surprised, but shocked when I did the analysis of the semen, which All India Coordinated Program project for bulls was being done. I analyzed the semen from the bulls, and I found that the fertility was from 3% to about 50%. Even 3% fertile bull was being used. Now, all the things, all the curse of it was not with that bull, but it used to go to that female. It used to go to that field. It used to go to that herd. It used to go to that farmer that, oh, it's female is not functioning. So please see that when we are talking of these principles of immunology, we apply them. And particularly these days, that with respect to the bull, with bull fertility, there are technologies which are now emerging, and no semen out of a laboratory should come out unless and until it is properly evaluated for its fertilization. My thanks to the organizers, to the university, and special thanks to those people who have been bearing me out and listening to me this presentation. Thank you very much. Pleasure talking to you all. Thanks.
Thank you very much, sir, Madan sir. It was a very nice, elaborative, and very fruitful lecture regarding the immunological issues and advancement in the fertility. And it is very beneficial to not only the academician, but also for the researcher. Your every single words are to be used for that. Now, if there are any queries by the some any attendees or participant, if you have any queries or questions, please raise your queries. Then we will further move to the.
Hello. Hello.
Yes, ma'am, please.
Yeah, good morning. Good morning to everybody. Honorable and very respectable our teacher of teachers and mentor of mentors, Professor M.L. Madan. Thank you so much, sir, for sharing a very, very good lecture. It is always good to hear you over and over again. And I enjoyed every slide of it, and each slide really made us all visit and revisit and learn. So, very humble thanks to Honorable Dr. Madan from my side. And I'm sure the people who have been listening would not differ with my opinion.
Oh, Taru, it's always kind of you to speak like that. Thank
Not
you
a
very
lot,
much.
not at all, sir. I think you would hear maybe better than this from others, because some of the people who must have heard you or who have heard you limited, they must be mesmerized, I'm sure. So, sir, it is really true, and for the benefit of the students who are here, I'm sure how easily sir has. Traveled right from hypothalamus, pituitary down the gonads, and how nicely sir could explain the antigen antibody reaction and talking about, when the interferon tau comes, it talks and it shuts the OT receptors and doesn't allow PGF2 to go. To all dear participants and all the respectable panelists who are here, and especially very seniors who are present over here. Sir, it was a very, very, very simple way you explained to everybody out here. I'm sure the students who are hearing, this is very important, that we understand what exactly happens, but how E2 receptors are being blocked, how the PG is shut, and what is the corpora lutea doing, and it's not allowing the OT receptors to take up anything, and then the whole cascading of pregnancy. And definitely it is a robust immunological phenomena. It is a question in front of all of us, that how does pregnancy sets in, and what sir has explained so simply, making a complex thing in a very simple way. I'm sure, sir, the participants and especially students and younger colleagues must have been benefited. I'm certainly benefited, and it's always, always good to listen. And a very clear diagram, simple yet clear, making complex things very easy. And that's a task of a teacher, which you have been teaching all of us. And thank you so much for that. And I really thank the Junagadh University having allowed me to witness this morning's Professor Madan's lecture. Because when I received the link only for day 2, I was thinking I'll miss this whole thing. I'm looking forward for the next lecture as well. So with that, I'm done. Thank you for giving me chance to speak. Thank you so much.
Thank you very much, madam. We are also very eager to hear from the Madan sir. If have any more questions to Madan sir, then please have any query. Then otherwise we move to the further next lecture by Dr. Kawani sir.
So we may clap for sir at least. Thank you so much, sir. Thank you very much.
Thank you very much, sir, Madan sir. It was very nice to see and listen your lecture. Your words are very important for us. Every physiologist, not only physiologists, every subjects, researchers, academicians have a lots mean of every word. Thank you very much, sir. So now we have another lecture that is a topic on the overview of the fertility in domestic animals by Dr. F.S. Kawani sir. Just have a before the start the lecture, I have some brief introductions regarding Dr. F.S. Kawani sir. Dr. F.S. Kawani sir had did his BVSc in 1971, MBSc in 1977, and did his PhD in 1985. He has been awarded with the Society ISSAR Fellowship Award in the 1996. Also, he received the Sardar Patel Research Award for the best research paper, and he has a very huge teaching experience, about 39 years. He also guiding the many students, 8 PhD students and 15 MBA students, and having 33 years experience of the PG teaching. And also, he had worked with the different schemes of Gujarat government, university, like cattle fertility scheme, non-plant scheme, then frozen cement scheme, RAM, the NATP improving reproductive efficiency of cattle and buffalo. And he has lots many, about more than 90 publications from international, in the international and national journals are there. With this brief introductions, over to you, Kawani sir. Please have a lecture. I invite Dr. Kawani sir to please deliver a lecture.
Namaste, Kawani sir.
Namaste, namaste.
Namaste, sir.
Namaskar, sir. Good morning.
Namaskar, very. Thank you for introduction.
Sir, Pailagu sir, Dr. Sudhar.
Oh, Sudhar bhai, how are you?
Thank you, sir. Fine, sir.
Good morning, good morning.
Hello, Sudhar.
Share the screen. Okay. Hello, are you able to see our screen?
Hello. Hello.
Yes, sir. We can see the screen. We can see the screen.
Are you able to see the slides also?
Yeah, we can.
Okay, fine, fine, fine.
I can slide in.
It is a second. It is a second. Just slides so करिए. ठीक है ना, पेज अप एंड डाउन એટલે થશે. ઓકે. માઉસ જોડેલ નથી ને એટલે આપણે એથી કરવું પડશે.
Okay, welcome you all. We know that India stood first in milk production since last 22 years. The total born population in India is nearly 300 million. And out of these 100 million breedable females are there. And they produce nearly 180 million tons per year. So per cow, our production is 1.8 tons It is very low compared to the developed country. If we see the dry wet ratio of our animal, breedable animals, they are nearly 60-40%, 60 and 40%. That means our 60% of animals suffering from either short or long-term infertility problems. And only 40% animals, they are regular cover. So, and that's why our production is still very low. We are using different terminology in animal reproduction, like fertility, infertility, sterility, and fecundity, etc. So the fertility means ability to reproduce. And if we want to define the term fertility, it is ability to mate, capacity to conceive and nourish the embryo, and finally expel the viable calf and fetal membrane. Infertility means it is a reduced degree of to reproduce. While sterility, it is very important. No doubt, the number of sterile animals are not more than 10%, but they are totally burden to our dairy farmers. As I told you, 300 million bovine population is there. Out of these, 200 million cattle and 100 million of buffaloes. And from this, our 100 million breedable females are there. Hardly 25 to 30% of these animals covered under artificial insemination. We know at present also the fertility rate under artificial insemination is not more than 50%. Even in buffaloes, it is hardly 40% or so. Our milk production is 175 billion tons. So per breedable female, we produce 1.75 tons of milk per year. In Gujarat, total breedable bovine population is 10 millions and the milk production is nearly 14.50 million tons. Now, overall incidence of bovine infertility is 5 to 40%. Now, 40% breedable females are regular calves, as we discussed earlier, and 60% animals having the longer intercalving periods. So, these animals are very important because they are suffering from the infertility problems. We do not know the actual cause of infertility in many animals. Out of this 60% animals, 10% animals are sterile, 30% animals are difficult to settle, and 60% animals having the normal body score, genital organs, but they do not conceive. This part is very important because there our managemental causes are existing in these animals to make the animals pregnant. Now we know that economic losses due to infertility, they are due to loss of milk production, loss due to the maintenance of infertile animals, loss of calf crop, replacement cost, and 15 to 20% bovines are culled annually for reproductive problems, just next to production problems. Now, if you see the minimum production days lost due to the major infertility problems, postpartum meniscus. Say this is the major problems in our animals. As animal cow, the farmers' total attention goes toward the production, milk production only. They do not see for the further, say, breeding of animal. So, if animal doesn't return to estrus or doesn't become pregnant within 100 days, then you can say it is a postpartum meniscus conditions. So, we have to give more attention to after pasteurization toward these animals. Post-service meniscus. Say here, pregnancy diagnosis is very important. Say Actually, we should carry out the early pregnancy diagnosis by progesterone and other tests, laboratory tests. But by manually also, by rectal examination also, one can diagnose the pregnancy as early as 40, 42 days or so. So we can say recognize the non-pregnant animal. Say here, the after service identification of non-pregnancy is more important than the pregnant animals, because non-pregnant animal we have to find out the what is the say reproductive status of the animal, so we can find out the causes of infertility in this animal, and we can prevent the post-service anesthesia condition and make the animal initiate the estrus and make the animal pregnant. Say repeat breeding, we lost nearly 63 days of production, and in abortion, we lost more than 108 days production. Say modern feeding and managemental practices on large scale are helpful in resolving this issue, but still challenges are prevalent. Now we know the established norms of the fertility. Age of first breeding should be 18 to 24 months. Say in our indigenous animal, it may be up to 36 months or so. Age of first calving is same way in crossbred animals and exotic animals, 30 to 36 months. In our indigenous cattle, it may up to 48 months or so more. Intercalving period should be 12 months in cattle and 14 months in buffaloes. First service conception rate should be 60%. Overall conception rate should be 80 to 90%. Number of AI per conception should not be more than 1.7. Calving to conception period should be, that means within 60 to 90 days after calving, animal should become the pregnant. The infertility problem less than 10% and abortion should be less than 3%. Now, with respect to bovine infertility, reproductive management should be aimed at the production of more milk with same number of cows, or production of same amount of milk with fewer high producing cows, increasing number of cows born per cow, decreasing number of reproductive culls, and reducing cost of producing the high genetic merit cow using artificial insemination. Say this requires regular and accurate evaluation of fertility, status of dairy herd as essential part of the sexual health control program at least twice in a year. Now, if we break up the major causes of bovine infertility under the field conditions, say nutritional and managemental causes more than 65%. Actually, this is in our hand. We can improve upon this lot, but the farmers, they are not taking care, because whatever they are having, they feed the animals. So the undernourished animals are more, and that's why the cause of infertility, particularly the nutritional and managemental causes, is more than 65%. Functional and endocrine disturbances are nearly 15%. Infectious causes 10 to 15%, and anatomical and hereditary causes are 1 to 2%. Now, if we see the practical point of view, three individuals are most important to manage the reproductive problem in the field. One is farmer, second is the, say, inseminator. It may be veterinarians, lay inseminator, livestock inspector. Third is the veterinarians. See, we see the poor management in aspect of nutrition at farmers' doorstep. The heat detection is very poor. Various stress factors are there, particularly the environmental stress, the unhealthy, unhygienic conditions surrounding the animals, poor hygienic conditions, and majority of the farmers, they are not keeping the record. Now, at breeding levels, we see the incorrect timing of air. say this is very important. First is the error in detection of the stress at farmer's level. He may bring the animal at any time, either in the beginning of the stress or many times in the exact timing of mid-stress period, or sometimes late or at the late state of the stress. And that's why the correct timing is not maintained and the conception rate goes down. Second point is very important. The wrong placement of semen dose. Actually, semen dose should be placed in just anterior to mid-cervical region. But many times we have seen that so either 1 or 2% semen insemination is carried out. You are surprised that even in the bladder, the AI gun may easily pass into the urethra and enter into the bladder. But the proper semen or fornix vagina, many semen doses is deposited in the fornix vagina. So the placement of semen dose is very important in artificial insemination, because we know that in natural service, semen is spray over the external os, but semen concentration is very high, and millions of sperm can enter into the cervical canal from the external os. And say in artificial insemination, only 15 to 20 million sperms are there, and if they are not placed properly, if it is placed in the fornix vagina or into the vagina, then may not be a single sperm enter into the, say, cervical canal. So it should be placed just mid-cervical region, because here we find the mucus is there, cervical mucus is there, which also bactericidal effect. So it may prevent the growth of the bacteria, or it may kill some of the bacteria entering into the female genital tract and protect the sperm. Now, the third point is very important, breeding hygiene. I have seen the many inseminators, they do not take care of cleaning the, say, actually posterior part of the perineal region of the animal which is being inseminated. And that's why the either urine or dung particles enter along with the, say, semen doses into the female genital tract, may lead to the genital infection. Then the lack of monitoring AI work. This is very important. Say, we fully trust over the AI workers, lay inseminators. We do not monitor them. We do not see what results pregnancy results they are getting. And if we monitor strictly to the artificial insemination work, then the better conception rate is expected from the AI work. At present, the conception rate is hardly 40–50% through artificial insemination. Now, at field level, we find many problems of diagnosis in infertility problems. Now, first of all, veterinarians should be able to judge the reproductive status of the animal at the time of examination, rectal examination. Say, many veterinarians in the field, they do not know the physiology of the, say, estrus and estrous cycle, or say the changes occurring during the different stages of estrous cycle in the genital tract, and they do not able to judge the proper stage of the estrus by rectal examination. Actually, we should not palpate the ovary at the time of insemination, because there are chances of rupturing of the follicle. But one should, in programmatic cases, one should go the thorough examination of the full length of the genital tract, including the ovary, and we should be able to palpate the functional structure, particularly the follicle and corpus luteum at the time of insemination. Say, early pregnancy diagnosis is also essential, and veterinarian should be able to carry out the pregnancy, he should be able to recognize the pregnancy at 40 to 45 days gestation period, to avoid the insemination in pregnant animals, because it was seen that 5 to 10% of animals may show the gestational stress during this period, and animal owner bring the animal for insemination. If the inseminator, the veterinarians may not able to diagnose the pregnancy at this stage, at early stage of gestation period, then there are all the chances that he may inseminate the animals, he may try to pass the AI1 into the cervical canal and disturb the say mucus plug of the pregnancy, and thus cause the early embryonic death or say abortion at this stage. So early pregnancy diagnosis, say every inseminator, or we can say the persons who inseminate the animals, either he may be lay inseminators, livestock inspector, or veterinarians, he should able to diagnose the pregnancy before he inseminate. He should say declare the animal non-pregnant before inseminate the animals, because it is very essential, because nearly 5 to 10% animals show some sign of stress. Even I have seen the animals showing the stress discharge, bellowing, uneasiness during early pregnancy, and owner think that each animal is in stress, and they take the animal for breeding, and say if he takes the animal for natural service, naturally seeing the sign of stress, animal is under the estrogenic effect. So it will also may give service, but as the semen is deposited in the say fornix vagina, naturally the cervical plug or pregnancy plug may not disturb. But as in insemination, we try to pass the pipette in the mid-cervical region, we disturb the pregnancy plug, cervical mucus plug, and may induce the embryonic death and abortion. Now, we should not leave the animal after insemination. The proper follow-up, either for the next subsequent stress, should be done. We should instruct the farmers that if animal return into stress after 20-21 days or up to 30 days, you should bring it again for the examination, or say if animal doesn't return to stress up to 2 months and more than 2 months, you should bring for the pregnancy diagnosis. This is very important, because it is known that nearly more than 30%, 35% animals after breeding, they do not return to estrus even though they are non-pregnant. And farmers think that each animal is pregnant. So the pregnancy diagnosis is very important. That is also as early as possible. Say one can diagnose the pregnancy by rectal examination after 40 to 45 days of, or go for the progesterone test in the laboratory and carry out the early pregnancy diagnosis to declare the animal, say, pregnant. Or say after breeding, to detect the non-pregnant animal is more important rather than the pregnant animal. Because if we do not detect the non-pregnant animal, the intercalving period will be increased. So we do not worry about the pregnant animal, but non-pregnant animal should be detected within 2 months of breeding, and such animal should be diagnosed for the problem, whether animal is enter into the NHS animal having the other infertility problems. That's why the pregnancy diagnosis is very important, and accordingly, finding the condition, we should manage the animal and bring the animal into the cyclic condition and for early breeding. So follow-up program is very necessary for maintaining the reproductive efficiency. And for this, a proper extension work is necessary. It is for the farmers, lay inseminators, even for the veterinarians is also important. Now, at present, there are certain emerging fertility problem when we implement the artificial insemination in large scale. We know that we have accepted the AI as a breeding tool widely. And from our herd, or say males are completely herd eliminated. So due to the absence of male, the certain problems are arised, like incidence of anestrus condition. It is increasing day by day. And this may be due to lack of male effect. And stressful climate, it may be the environmental conditions or high milk productions, etc. And also disease factors. So presence of male augments the reproductive activity of females mediated through the hormonal and neural mechanism arising from the action of male pheromones and sexual interactions. We also find high incidence of subestrus condition. That means animal in estrus but not showing the behavioral sign of estrus. So it may be both to micronutrient deficiency, stress factors, but the male effect is very important because it is the duty of male to detect the stress animals. So biologically, heat detection is responsibility of the male animal for mating to take place at right time. Third is the ascending genital infection. Here, the poor breeding hygiene and calving hygiene is very important. If we observe the calving in village, it is generally, say, in unhygienic condition. And also, the breeding carried out at most of the AI centers, the lay inseminator, they do not take care the cleaning the perineal region of the female animals, and they inseminate the dung particles, urine droplets along with the semen. They put deposit these unhealthy materials into the female genital tract. It is the presence of male has been found to make heat sign more pronounced in females, and hence the disappearance of the behavioral signs during heat is attributed to lack of interaction with male and maintain male experience in past. Now, we also found that the normal heat period is extending. Normally, we find that the duration of estrus in cattle and buffaloes, it is nearly 18 to 24 hours.
भारत
But
में लोग सर क्रांति सर इसमें तो आती नहीं क्रांति का महाराज जी होता क्या नहीं
म्यूट करोगे તમારો ઓકે સુથાર બોલે म्यूट કરોगे તમારો ચાલો આપો આગળ.
So, normal duration of estrus is 18 to 24 hours in cattle and buffaloes. But in many animals, we find lengthening of the estrus period. It lengthens up to the 48 to 72 hours in our animals. So, rapid cessation of heat sign following mating has already been proved in animals like dogs and goats. Mating causes earlier arrest of heat sign in bovine than unmated ones. Prolongation of heat may be due to lack of mating stimuli. Stimulation of clitoris following artificial insemination has been found to shorten the duration of heat. We also found the high incidence of delayed and anovulations. Mating stimulation is necessary for ovulation in cat, camel, rabbit. They are induced ovulators. In farm animals, they are spontaneous ovulators. What is the male effect? Say, actually, initiation of reproductive activity in female, induction of pronounced heat sign. In presence of male, usually the female shows the pronounced heat sign. Accurate detection of heat is the duty of the male. It helps in induction of ovulation. If we go the general classification of the causes of bovine infertility, so is say it is anatomical, like hereditary and acquired. Hereditary, no doubt, the incidence is very low, but it is important because they are totally sterile animal. The condition like ovarian agenesis, premartinism, and uh, hermaphrodite animals, they should be identified as early as possible because they are totally burdened to the farmers and they should be eliminated. The functional form of infertility, it is mainly due to the hormonal imbalance or dietary and excess. Say actually in this condition, we find cystic ovarian degeneration, delayed ovulation, annulation condition, etc. Say the more important is delayed and annulation condition because these animals, they come into the estrus, we breed such animals, but as the ovulatory functions are disturbed, so many animals, they do not ovulate or there may be delayed in ovulation, though there is no conception. So this animal should be identified and they treated accordingly. The infectious disease, specific and non-specific disease, they are still existing in our animals. Nutritional deficiency, very important. Mostly our animal, they are deficient in, say, nutrition, either it may be energy or mineral deficiency, or a few animals, dairy animals, which are, say, high producing or in organized farm, there may be excess of nutrition leads to infertility. The majority of the part in managemental infertility includes the incorrect record keeping, problem with heat detections, semen quality, incorrect timing of PI, very important, stress factors, it may be the physical stress, environmental stress, production stress, and lack of exercise. Now, preventive measures to minimize the bovine infertility. Say, actually, after parturition, the attention of the dairy farmers goes toward the production. He is interested in milk production only and do not think of rebreeding his animal. So if animal doesn't come into the estrus, even after 90 days in lactating animal, the farmers do not worry. And that's why the intercalving period is increasing more than normal and animal enter into the infertility conditions. So parturient cow and buffalo should be examined between 50 and 60 days after calving to assess the status of reproductive system. At this stage, actually, animal may be in true estrus, many animals may be entered into the cyclicity, some animal may having the, say, genital infection. Accordingly, we have to, say, manage the conditions. Depending upon the finding of estrus cycle should be induced in the anestrus animal and efforts should be made to make the such animal pregnant within 100 days post-calving. So we can maintain the intercalving period 12 to 13 months in dairy cows and 14 to 15 months in buffaloes. The barn breeding and calving hygiene should be maintained. This is very important. Say majority of the dairy animals, they are kept in very unhealthy barn hygienes. The stable hygiene is very unhealthy. We are going to see the few pictures of unhealthy barn hygiene. At breeding also, at artificial insemination stage, say at this condition, I have seen many lay inseminators in dairy cooperative areas. They never clean the perineal region before insemination. Many times the dung particles, urine drops adherent to the AI gun at the time of breeding. So they entering the infection rather than insert semen into the female genital tract at the time of breeding. Same way, the calving hygiene is also at farmer's doorstep. In majority of the farmers, they do not take care of calving hygiene. Calving hygiene is, say, very poor, unhealthy, and dirty place. So it should be, say, one should take care of barn breeding and calving hygiene. It is very important because it may lead to the post-breeding or post-calving genital infection. Say, strict monitoring of AI activity and refresher training for AI workers is very necessary because many of our AI workers, lay inseminators, they are trained for 15 days or 20 days and enhance the duty of the AI work. But they do not know the proper physiology, they do not know the pregnancy diagnosis, they do not able to diagnose the, say, whether animal in estrus or not in estrus or having the, say, infection, genital infection, and they are totally, majority of the AI workers totally depends upon the history provided by the farmers or dairy men, and they inseminate the animals. Even I have seen the animals inseminated during infection, uterine infection, or say in any stress condition, in early stress of the stage, in pro-stress condition. So the monitoring of AI activity is very necessary, and this is the one of the major cause for lower rate of insemination or lower rate of conception by insemination in our dairy cooperative areas. Now, breeding hygiene. It is directly related with animal health, protection, and fighting infectious disease. Veterinarian must know the prevention system, biosecurity measures, and animal welfare issues. Farming and implementation of regulation for animals' health protection, production, and maintenance are required. Now, in the field, we have seen the very poor breeding hygiene when at the place when the insemination being performed. We know that inseminator is a very key person in animal breeding system. And in majority of the insemination are performed in unhygienic surrounding conditions. Instead of we should say they carry out the artificial infection rather than the artificial insemination in unhygienic conditions. So inseminators must be sincere enough to follow the hygienic in every possible way and must follow the scientific methodology. He should be educate the farmers to regarding the cleaning of animal before we bring the animal for insemination. Particularly the perineal region should be cleaned before insemination is performed. He should be sufficiently skilled and experienced to diagnose the estrus, early pregnancy, and pathology conditions. Say, we cannot expect this thing from our lay inseminator. Our 90% insemination in the field are being performed by lay inseminator. Even they are not able to judge the estrus. In many conditions, they are failed to judge whether animal is estrus or not. Forget regarding the estrus stage, whether animal in early estrus, mid estrus, or late estrus. So we should train our inseminator for diagnosis of proper estrus or stage of the estrus. He should be able to raise the doubt regarding the early pregnancy because you have seen that 10 to 15% early pregnant animals show some sign of pregnancy, some sign of estrus. like bellowing, even sometimes uterine discharge, and owner think that his animal is in estrus and he take for the insemination. And at this stage, if inseminator doesn't know the pregnancy diagnosis, then there are all the chances that he may inseminate the animals and it may cause the embryonic death or abortions. Say, many inseminators, even they do not able to recognize the pathology of the reproductive system, particularly the in uh, endometritis, unhealthy discharge. They think that it is stress discharge and go for insemination. See, same way the cowing hygiene is very important. The majority of our insemination, cowing in the field, say cowing season, particularly in August, September, November, December, which is monsoon seasons, and the condition of the dairy animals at farmers' doorstep is very unhygienic. So, and majority of the animals they cowed in unhygienic conditions, unhygienic surroundings. So, in nearly 90% animal get reproductive contamination during calving. Out of this, say many of these bacteria are opportunistic, and about 25 to 30% animals will show the sign of metritis. See, out of these 30% animals, within 3 weeks, about 15% of dairy cattle developing the sign of clinical endometritis. And this is due to poor hygienic condition or poor assistance provided to the animal by the farmers or by the, say, livestock inspector or veterinarians in unhygienic way. It may lead to the infection, and infection may persist in 15% animals which develop the endometritis after calving. And this animal take more time to conceive. They enter into the infertility problems in future. Thus, the endometritis is an expensive condition for veterinarians, farmers to manage like a costly mastitis. We know that the infection disturbs the endometrium and luteolysis. It disturbs the function of the prostaglandins. And also, bacterial toxin binds directly to the uterine epithelium and stromal cells and stimulate the release of PGE2, which is luteotrophic, and many may prevent the luteolysis. Bacterial endotoxin and cytotoxin suppress the release of gonadotrophic releasing hormones from the hypothalamus and disturb the secretion of LH from the pituitary gland. The net effect is that there is a failure of ovulation, increase the cystic ovarian conditions, and anovulatory stress. Now, at present date, subclinical endometritis is very important. Here, there is no clinical signs. Animal coming into estrus, there is no unhealthy discharge from the genital tract, but it persists the infections. And it significantly reduction in reproductive performance. It can be diagnosed by cytological studies. Risk factors associated with the uterine infections is abortion, dystocia, twins, retail paper membranes, and metabolic disease. Control of endometritis focus on optimizing animal health by paying the attention to barn, breeding, and calving hygiene and feeding practices. A negative energy balance or deficiencies of micronutrients via selenium, vitamin E can suppress the immunity. Now, personal hygiene is also very important. Effective biosecurity program must be addressed the potential spread of disease by individual within the and between the farm having the direct or indirect contact with animals, their excretions and secretions. People and vehicle coming in direct contact with the animals must be limited along with use and frequent change of protective clothing, footwear use, and sanitizers and farm sanitizers. Now we come to the biosecurity plans. So this is very important nowadays. Biosecurity plan means control and avoid introduction and spread of infectious agents in the farm. It is a part of health management program. Proper use of set of working instructions and protocols. How to build up a biosecurity plan? Make the farm more closed. Design a people entrance protocol. Design an animal entry-exit protocol. Determine on farm unit and section with the respective protocols. Set the protocol for healthcare, feeding management, and hygiene and disinfectant. Design an on-farm monitoring protocol. Instruct the farm workers, assign the responsibilities, and evaluate their performance. Define a farm-specific training program for the farm workers in the different units. Critically evaluate the risk factors periodically. Regular team assessment of protocol for possible adjustment in biosecurity plans. Now, you see the biosecurity plans to secure the life and protect from the disease. Management to practice to protect the herd from the disease or protect from the introducing the new disease. See, it helps in milk performance, maintenance the normal reproduction, necessary for food safety, longevity, animal well-being, ethical aspect, and economic. Now, biosecurity may be for particular animals, it may expand to the unit of the animals or in the whole farm, particular region or country. Step to prevent the transmission. Quarantine or isolation stamping is necessary. Restrict the access to the farm. Reduce the traffic to specific area. Routine to wash and disinfect equipment and vehicle. Safe feed and water supply. Safe and efficient manure handling. Now, these are the certain pictures like clean vehicle and clean equipments, cleaning and disinfecting the farm. Say, a ideal barn, see the proper sunlight, protection from the rain and extreme heat, proper ventilations. Here is the village conditions, the animal are kept like this in dairy cooperative areas in Gujarat. These are the conditions. Say in very unhealthy condition, animal are kept. The animal shed is also not as per the standards. Very unhygienic conditions. The animal tied with short rope and in muddy areas. Say one should take extreme care while performing the insemination. We know the placement of semen is very important. It should be placed in the just anterior to the near the body of the uterus during insemination. Evaluation of the semen, the loading of AI guns, cleaning of the perineal regions, and the external genital organs of the cow before insemination, introducing of the AI gun, calming in unhygienic conditions, the hygienic score for the udder. Here some score 2 is little dirty, score 3 is unhygienic conditions, score 3 and score 4. Here the dirty, stable, congested, stable. Animal is drinking unhealthy waters, dirty and muddy areas. Animal are tied very shortly. Bathing and grooming is also necessary. Here is some ideal, stable, cleaning of udder teats before milking. Mustn't milking, but 5 minutes. Cleaning and inspection of the animals. These are the high cost technologies. Now, we show the farmers should educated for good management, hygienic and immunizations. Protection of the animals from the extreme weather conditions. Balanced feeding is necessary. This is our limitation. Majority of the dairy farmers, they feed whatever they are having, and there is a lot of seasonal variation in feeding. So the reproductive efficiency and milk production is affected. The critical heat detection is very important because it leads to timely breeding. The early pregnancy diagnosis is very important because if we do not carry out the pregnancy diagnosis at early stage, then it is good, say, okay for the pregnant animal, but if non-pregnant animals, they are not detected as early as, say, 45 days or so, then many of them, they enter into the, say, anxious condition, and farmers, as the animal doesn't return to estrus, they thought that this animal is pregnant, and they do not take for the pregnancy diagnosis, and later on, they find that the animal is not pregnant. So it is very important the pregnancy diagnosis should be carried out at as early as possible, before 60 days or so. The supplementation of minerals, vitamins are necessary. Control of endo and ectoparasite is also very necessary. Record keeping. Majority of all dairy farmers, they do not keep the records, but it is very necessary for breeding and milk productions. Preconceived wrong belief, misconception should be removed by thorough extension work. So for successful implementation of rigid sexual control program, a large number of trained veterinarians, especially in the field of animal reproduction, are required. Thus, the veterinarians should be given periodical exposure to the technical know-how to solve the reproductive problems, opportunities, and agencies. Thank you all.
Very nice lecture, and it was very informative. I am sure your lecture is not only help to the students but also to the field veterinarian And here you have also elaborated how to diagnose the infertility in female and how to treat them. Also, sir, you have given what are the different preventive measurements are to be taken to improve the fertility. Uh, Now, I would like to invite any speaker, any attendee to have a question for on this topic.
Dami સાહેબ શું જુઓ છો?
ઓળખાણ શોધું છું કે ઓળખાણ.
Dr.
આ કોઈ.
Thank you very much, Dr. Tulk, that you have provided us the opportunity to listen my mentor and teachers. After I have first listened in 1983, and I am surprised that today also, with the equal zeal, with the same speech, with the same faith and everything, with the same enthusiasm, he taught us again once more. Thank you very much, sir, for that. Then sir has covered almost every aspects of the infertility. That is really a overview he has given. He has also included, sir, the recent approaches for the management and what is the today what is the need of today to improve the fertility of the animals. That also he has included. I'm surprised, sir, that even at this stage also the speech, the delivery, the lecture system, the pattern, everything is same as I have listened to you in the 1983-84, sir. Thank you very much, sir.
Okay, thank you very much.
Hello, Dr. B.N. Sudhar.
Yes, sir.
You are equally enthusiastic, looking so smart and involving in full lectures in spite of now being head of your department and all.
I have gone through the program and I could not miss the lectures given by Dr. Madan sir as well as my mentor and my guide Dr. Kawani sir. I can never miss that.
So we are very grateful
Both are legend in the field of animal reproduction.
Consistently attending the event.
Thank you. Thank you.
Any questions from the participants, students or faculty from other institutes, our own institute?
If there is no question, then we conclude today's first session of the lecture. We finished here, and before that, I would like to very much thankful to Dr. Madan sir and Taruman. They are continuously keeping touch with us on the screen. We are seeing all the other faculties from the different institutes are also bearing with us. And I'm very much thankful to our principal and Dean Dr. P. Tank sir, organizing and coordinating secretary are also continuously here with us. And so we conclude and finish the first session of the theory, and we will be meet at sharp 3 p.m. for the demonstration of the practical that is on the routine methods for the collection and evaluation of semen, and that is be delivered by Dr. G.B. Solanki, S.S. Parikh, and Dr. J.K. Chaudhary from the cattle breeding farm, Junagadh Agriculture University, Junagadh. So thank you everybody. Thank you very much.
Thank you, Dr. Scala and Dr. Arun. Bye for now. So, and regards to all the seniors out here. So bye for now.
Bye.
to avoid the injury. So these are some of the pictures which are taken by me during this semen collection. This is a chin resting in the bulls, which can be used in the scoring of the sexual behavior. This is a sniffing of the perineal area. This is a Fleming's reaction which is shown by the bull. This is a up curling of the upper lip and extending the neck. This is a penile erection. This is a penile protrusion in intromission into the artificial vagina. And this is the ejaculatory thrust. You can see that both the legs of the bull are lifted, both the rear legs of the bulls are lifted, and it's a typical cattle bull thrust. Now I would like to show some videos which are taken by me. For that also I have to share the screen. So this is a video of the collection of semen from the Gir bull. Normally we use the male animal as a dummy. So this is a Jafrabadi male bull which is used as a dummy. This is semen collection. Firstly, we try to give 1 or 2 false mounts to the bull so that it can get ready for the collection. So these are various aspects of the sexual behavior. The courtship, then there is a sexual arousal, there is a sniffing of the perineal area, the penile erection, penile protrusion from the prepuce, then intromission, and then ejaculatory thrust. This all comes under the reaction, then the refractory period, and this all comes under the reaction time. There is a time taken by a bull after the exposure to the dummy to the successful ejaculation. Some scientists again recommends that the reaction time can be taken as the bull, the exposure of the bull to the dummy to a first mount or first fall mount with the erected penis. See, this is the semen collector is getting ready to collect the bull, semen from the bull. The semen collection should always be done in a very clean, hygienic, and very calm place. No unnecessary noise should be made. So this is the intromission. This is ejaculation. See, again, you can see classical thrust from the Gir bull or from the cattle bull by lifting both his hind legs. And I will show one another video of collection from the Jafrabadi bull. These Jafrabadi bulls are more reluctant again as compared to the Gir bulls. They are shy breeders. The sexual behavior scoring is low. There is a erection of the penis. You can see here. There is a sniffing of the perineal area. So this is in Jafrabadi buffalo bulls. We use female animal as a dummy because if we use the male animals, then they try to fight each other and they are becoming very furious when they see another male animals nearby. So this is the first fault mount which is to be given. And there is again the erection of penis, and the bull is ready, getting ready to donate the semen. So the fall mounts, again, we can give 1, 2, or 3 accordingly when you know the sexual behavior of that particular bull. This is intromission and this is ejaculatory thrust. Normally in buffalo bulls, they do not jump and they do not lift both the hind limbs from the side. Rather, they only curve their posterior part, or you can say the back. So now this was all about the semen collection. Then we moved forward to the semen evaluation. So there are different tests to assess the sperm quality. That is the first is the microscopic examination. We go for microscopic examination right after there is the entry of the semen from that pass box. We go for the evaluate the volume microscopically. Then microscopic examinations, various biochemical tests, there are various resistance to environment tests, physical evaluation of the semen, chemical evaluation again, and various biological tests or the in vitro fertility test. So macroscopic evaluation, it contains the volume, the color, what is the color of the semen, it may be milky, it may be creamish, and the consistency or the density of the semen, whether there is any contamination, the presence of the foreign material or anything, and the gross motility in the microscope. So these are the average volume of the semen in different species. We are collecting semen from Gir bulls and the Jafrabadi bulls. So in Gir bulls, the range we are getting is nearly from 1 to 15 ml with the average of 5 to 6 5 ml. And again, the Jafrabadi bulls, the concentration is high and the volume is low. These are some of the nomenclature for different evaluation criteria. That is, if there is no sperm in the ejaculate, it is called aspermia. If there is a reduced number of spermatozoa, then hypospermia. If there increase in the number, then hyperspermia. This is when we see the volume and the consistency of the semen. Then color of the semen, again, it is very much important. It can be milky white, creamy, or opaque. Normally, buffalo semen is whitish when compared to the cattle bulls. It may vary from watery, thin milky, thick milky, and creamy. So these are some of the abnormalities or some of the abnormal colors which are found during the semen collection. This brownish in color, then there may be orchitis or there may be blood pigments in the semen, the dark red or the pink blood, then the hemorrhage in the male reproductive tract, morely this urethra and thing. Then this yellow green coloration is there. Then there is chances of the infection from pseudomonas. Light brown contamination with the drug, dull and dirty white. Then there is increased number of spermatogenic cells. Yellow, again, there is a presence of urine and chunk clots or the curdy appearance. It also indicates that there is an infection. The color of the semen in the cattle bulls, generally in the cattle bulls, it is more yellowish due to the presence of the pigment riboflavin, and in the buffalo bulls, it is white or milky white. This is a range of bulls. That is similar, typical watery semen with the very low concentration of spermatozoa. This is a typical good ejaculate. It is having a creamy consistency. Then note, you can see here various types of ejaculate. Here is a collection of the sand at the bottom when there is a contamination of the semen. There is here this aluminum foil get deposited into the semen. This is the fodder part or the hay, which is also due to contamination. There is a presence of the hay. Again, the viscosity, consistency, there is a difference in the consistency. It may be from the watery semen up to the creamy milky. And accordingly, the semen is to be graded according to the consistency. If it is thick, creamy, it is excellent. The concentration of the spermatozoa may be over 2000. It is creamy, then 1500 to 2000. Thin, creamy, then 100 to 1500. Milky, poor, 500 to 1000 millions per spermatozoa. And it is watery, then the grade is very poor, and we normally do not use it ejaculate for further processing. The concentration is below 500 million. And this is a microscopic examination which is to be carried out before further processing of the semen, which includes the mass activity, the individual activity, and the concentration estimation, so that the accurate number of doses which is to be prepared can be known, and the amount of diluter which is to be added, we get that idea. Live and dead sperm estimation, acrosomal and abnormal sperm count, and acrosomal integrity. And this is a mass activity. Here it is graded into from 0 to 5. I would like to show a video which is taken for mass activity. See here, you can see the typical swells and ADs, the waves which are seen in the sea. Here, this is a collective mass activity. This is a colored view. So we can see the rigorous movements of the spermatozoa, and accordingly, with the frequency or with the amplitude of these waves, or we can say the motion or the movements eddies, we can grade the semen from 0 to 5. Normally 4 plus and 5 plus semen are to be used. 3 plus can also be sometimes used. Then comes the individual or the progressive motility. There are 4 types of motility. Progressive motility, which is highly beneficial, and which is the progressively motile spermatozoa are used for further processing. The circular and jerky motility can also be seen. Oscillatory motility, the sperm do not move from its place, but rather oscillate on its place. And there can be sometimes this reverse motility. Again, a video of the individual motility. So here you can see the nearly all the spermatozoas appearing in the feed and getting out. That is in and out. They are progressive motile. Some of this spermatozoa are reversely motile. So the maximum the number of the spermatozoa in your ejaculate should be progressively motile. Then this is if the number of spermatozoa that is 90 to 100% are progressively motile, then it is graded as excellent quality ejaculate. If it is 70 to 80, it is good, and below that we are not using. If there is a cutoff that 70% of the spermatozoa should be progressively motile in a mid semen. And again, the sperm concentration, this is fresh semen, helps to determine the dilution rate, and also that the how much number of straws will be prepared. Again, estimation of sperm concentration, previously it was done by this hemocytometer. This hemocytometer, you know that which is used for the counting of RBCs and the WBCs, which can be used. The visual rough examination will give you the idea of the concentration of the semen. Colorimeter, and this spectrophotometer normally we are using, and it is used in all the semen stations. This nucleo counter, the computer assisted semen analysis. This is CASA again, which can be having a parameter that it measures the sperm concentration. This is flow cytometer. I'll show a video of the spectrophotometer. So this is a Hamilton Thorne photometer, which is used for the estimation of the sperm concentration. 40 microliter of semen is sucked into this cuvette. Then again 1 ml of the normal saline and the semen is given in this cuvette. We have to feed that what is the volume of the semen. Suppose ejaculate volume was 5 ml, 40 millions per spermatozoa. Then this is giving that it is 758 million spermatozoa per ml. This is a photograph of the nucleo counter, which was used previously for the estimation of the semen. Hello. This is a hemocytometer, which is used, which can be used for this standardization or of the photometer to check that yes, the readings which is given by photometer are correct. This is the normal sperm concentration. The cattle bull 1200, buffalo bull 800. The range is very much more. This is a nomenclature which are used. Normozoospermic, there is normal sperm concentration. If the concentration of the sperm is reduced, then it is oligozoospermia. Increased number of concentration, polyzoospermia, and zero sperm concentration is azoospermia. So these are the minimum criteria for the semen processing. The ejaculate should pass all the criteria. If the volume is 1 or more, we can use for further processing. The sperm count should be more than 500 million per ml or more. Motility of the progressive motility should be more than 60 or 70%. Total sperm abnormality should be 20% or less, and the intent appreciable integrity spermatozoa should be 70% or more. So for cryopreservation of semen, this extender plays a major role. The extender should be having the nutrient as a source of energy. It has to protect against the harmful effects of the rapid cooling, provide the buffer provide the buffer to maintain the osmotic pressure in the semen, and protect the sperm cell during the cryopreservation. So these are the principles of the sperm cryopreservations. The temperature, the energy source is in the form of sugar is to be given. Osmotic pressure in the semen should be maintained. Electrolyte balance should be maintained. The buffering and the pH is to be maintained. There should be no growth of the microorganism, and the semen or the spermatozoa should be protected from the cold shock. These are different approaches of the semen preservation and storage. Previously, they used collect the semen and then go for the insemination or other the storing at ambient temperature at 20 to minus 30 degrees Celsius. The duration we can use the semen was only 2 to 3 days. Again, some stations were using this chilled semen at a refrigerated temperature. Again, 2 to 3 days of duration we can use. Then this frozen semen, and lastly the deep frozen state, that is minus 196 degrees Celsius using liquid nitrogen. And for that, this semen is packed into 0.25 ml or 5 ml straws, and for after that, after the cryopreservation, we can use the semen for several years. So this Gris Agio fructose citric acid glycerol, it is known as the universal extender, which is mainly used in all the semen stations. These are some of the commercially available extenders which are available in the market. There are powder coconut water based, this Gris based are also available. Commercially egg yolk based medias are also available. Skim milk based lessipos. The soybean, again, the use of the egg yolk is causing a problem of various, that it is from the animal origin protein, so it should be avoided again. So this soybean or the plant source lecithin is used in some of the commercially available extenders. We are using this Andromed ready to use extender at our semen station. So this is the extended preparation. This is a Andromed bottle, 200 ml packing is available. We have to add 800 ml of water, that is 1:4 dilution rate. And so this is the pouring of the Andromed, then the water, and again the dilution is prepared. The diluter is prepared and it is kept at 34 degrees Celsius for further use. And again, this semen is collected. It is entry into the laboratory. A liquid is used for the evaluation, for the volume, for the mass activity, for the concentration evaluation. Then it is further initially diluted with the equal amount of the diluter. For example, there is a 5 ml of semen, and 5 ml of diluter is added, and it is kept in the water bath at 35 degrees Celsius. And the final dilution is done after the evaluation that the ejaculate should fulfill all the criteria which are must for processing. Then the diluted semen are then packed in the plastic straws that are 0.25 or 0.5 ml. I will show you a video of the vacuum. So this is a video of the filling sealing machine. We are having a MRS-1 filling sealing machine. It fills and seals one straw at a time. So see here from this is a diluted semen. It goes from here to the nozzle and it is sucked into the straw. This is another end from where the vacuum is applied, and this is a hammer by which the straws are sealed by using the pressure and also heat. This is from where the vacuum is created so that the semen is entered into the semen straws. This is a vacuum pump which is attached to the filling sealing machine. So this is at a single time operation, 100 straws are automatically sealed and filled and sealed. This is container. Then after filling and sealing, these straws are arranged on this rack in this manner for the vapor freezing. The 100 straws are arranged in this rack in this fashion. in this manner. Then again the second rack is applied over the ramp for another straws, another batch of straws. Then after arranging the racks on the after arranging the straws on the rack, they are kept for equilibration in this cold handling cabinet, which is kept at 4 degrees Celsius. The equilibration time is of 4 hours. And after keeping that the cement straws for 4 hours at 4 degrees Celsius, they are further used for vapor freezing. They are kept at 10 to 12 minutes above 4 centimeter of the liquid nitrogen vapor. Then they are finally plunged into the liquid nitrogen at minus 136 degrees Celsius. So here I will show again a video. See, we are not having this biofreezer, so we are using this conventional method of freezing. Here, this stand is kept in the this thermocol box, which is lined. That here, this is a line, and the liquid nitrogen is filled 3 centimeter below that stand. This rack from the cold handling cabinet are collected, and they are kept 4 centimeter above the layer of LN2 for 12 minutes. And then after 12 minutes, we collect the straws in this manner. The temperature goes from 4 degrees Celsius of the equilibration cold handling cabinet to minus 140 degrees Celsius by this vapor freezing. And after minus 140 degrees Celsius, they are plunged into the liquid nitrogen, and the temperature goes to minus 196 degrees Celsius. And these empty racks are taken out, and again the racks with the nowadays this IOS2 filling sealing machines, automatic filling sealing machines integrated with the printer are also available. I would like to show a video. It was shared by one of my friends. This is a machine. We have to take a program. The ejaculate number, it can be 1 or 2. These are the details which are to be printed on the cement straws. Then they push the button go. And this is the IS4 filling sealing machine. This is a diluter chamber. So in this machine at a time, 4 cement straws are filled and sealed. You can see here. So this type of machines are used in the cement stations, which where the production unit is very much higher. So this is a printed automatic printer is integrated in this filling sealing machine, and the straws get printed. Then these are some of the programmable biofreezers which are used nowadays in the established cement stations. These are having capacity of 50 to 100 cement straws which are vapor freezed at a time. This is the racks which are arranged in the vapor freezer. This is a tank which is attached with the biofreezer, programmable biofreezer. And this is in this programmable biofreezer, there is a standardized graph which is prepared, and the freezing rate is very accurate. So for example, this is first is for a minus 10 degrees up to minus 10 degrees Celsius, the freezing or the cooling rate should be 5 degree per second. Then in the second up to minus 10 to minus 100, the freezing rate or the cooling rate should be 40 degrees per second. Then in the wrap minus 142, again up to minus 140 degrees Celsius, the cooling rate should be 20 seconds. This is a program which is to be run for biofreezer. This is a picture of the biofreezer. Then again, for the large production cement straw producing centers, they use and they collect the straws in this manner by keeping the goblets in the wide mouth liquid nitrogen container. And again, the throwing is very much important. Throwing is to be done at 37 degrees Celsius for 30 seconds to evaluate the quality of the post-throw cement. Here are some of the important points which are to be kept. The time to throw with the watch to avoid the guessing. There should be no guessing. Then the post-throw evaluation, the primary parameter which is used from long time is the post-throw motility. We have to go for the post-throw motility evaluation. I will show a video of the post-throw motility. This is a video of the post-throw motility. Normally, more than 50% of the spermatozoa should be progressively motile. If it is less than 50%, then we have to discard that particular batch of cement straws. See, there are various dead cells or the cells which are moving in backward direction. The sperms only having the oscillatory movements. So the cutoff is 50% post-throw motility for the successful artificial insemination. Then the live dead evaluation of the spermatozoa, there are various methods. Normally, we use this eosine, nigrosin, or the vital stain for this evaluation of the live and dead spermatozoa. There are various, again, fluorescent dyes which are used for the evaluation of the liability of the spermatozoa. The host 33342, DAPI, propadium iodide, CFDA, the cyber 14, and the host assay. So this is a method you might be knowing. This how this smear is prepared for the live and dead estimation. This is a thick smear, and these are the some of the live and dead spermatozoa. The spermatozoa which are dead, the stain gets inside of the plasma membrane, and they take the stain, so they appear pink on the screen. And the live spermatozoa which are having the intact plasma membrane, they do not take the stains, and so they appear white. See, you can see here the B spermatozoa which are pink in color have taken the stain and they are dead, and these white ones are live. This is the again the live and dead spermatozoa by using this fluorescent dye. Again, these illuminating cells are alive, and these dead cells are dark. Again, here live, which is illuminating, and this red one is dead. This is the normal spermatozoa structure that because we are going to see some of the sperm abnormalities. So this is the normal structure of the spermatozoa. This is the head, this is midpiece, tail. In the head, there is a acrosome and the acrosomal cap. The nucleus is inside the hell. This is inside the head. This is the mitochondria or the midpiece, and then again the tail. So these are some of the sperm abnormalities which are seen after the cryopreservation. When you see the head defects, then detached head, there's a large head, small head, elongated, small acrosome, no acrosome, the acrosome get eliminated, vacuolated spermatozoa or the irregular acrosomal cap. In the neck and the midpiece, there is asymmetry of the midpiece, bent midpiece, thin midpiece, thick midpiece, irregular or the corkscrew, and the cytoplasmic droplet may be proximal or distal. And tail defects, the coiling of the tail, short, this is a hairpin shape, the stiffening of the tail, this is broken tail, two tails or duplicate tail, and the terminal droplet. So this is the slide showing the double head or the double tail. This is here, the spermatozoa are having the double head, and this is also double head. This is the double tail spermatozoa. Then this is a diadem defect. You can see there are spots of in the form of coronation in the postnuclear region. and underdeveloped, or the teratoids, which are also seen sometimes in the semen. This is narrow at the base. The base of the place where the midpiece is starting is very much narrow. You can see here, it is not flat. An abnormal contour of the spermatozoas are also seen sometimes. The corkscrew shaping of the midpiece, here this is seen like this. And micro head, the head is small in comparison to the normal cell, and this is again a small abnormal head. The pear shaping of the head, normally it should be like this. Again here the pear shape of the head. And the proximal doublet, which is again a sign of the maturational defect. This is the proximal doublet, you can see here. This is proximal doublet. And the pseudo doublet sometimes are also seen in the midpiece. This is a midpiece defect. There is a tail stump is there, short or a very short midpiece. That is segmental aplasia sometimes are also seen. And this is abaxial implantation or abaxial attachment of the midpiece in the tail. It is not symmetrical. And there's a coiling of the tail from the terminal end. This is again the distal protoplasmic droplet. This is the DAG defect. There is a curling or the tightening of the midpiece or the tail over the midpiece. You can see here. And after the sperm abnormalities, we move toward the assessment of the acrosomal integrity. So the see this acrosome is here, and this acrosomal cap contains the various enzymes like acrosine and hyaluronidase, which are very much important during the penetration of the spermatozoa into the ovum. So this is to be this acrosomal cap is very much vital, and it should be maintained for the proper or for the fertilization of the cell. So you can see here this acrosomal cap very well maintained. So it is called the acrosomal intact spermatozoa. And there are various types of acrosomal abnormalities. For example, nobed acrosome, ruffled acrosome. There is a loss of acrosomal cap, detached or disintegrated. This is a intact acrosome. You can see here there is a clear-cut acrosome is seen, and this is a knobbed acrosome. There is a knobbing here. This is a lost acrosome, completely denuded spermatozoa, or the acrosomal cap is lost. See again, there is a ruffledness, or the ruffled acrosome is there. The integrity of the acrosomal cap is lost. And this is again the detached acrosome. Here, this is a detached Gilia capitis, or the acrosomal cap, which is seen in the slide. Then we move towards the host. Host test, or hypoosmotic swelling test, which is used to evaluate the plasma membrane or the intactness of the plasma membrane of the spermatozoa. So the basic principle for of the host test is the active spermatozoa exposed to the hypoosmotic or the hypotonic solution, and there is an influx of fluid from the exterior to the interior of the spermatozoa. So there is a if the plasma membrane is intact, then there will be swelling of the spermatozoa and also the curling of the tail. So this is the host test. This is a host positive spermatozoa. You can see there is a thickening of the spermatozoa of the tail, and also there is a curling of the tail. These are the B. These are non-host reactive spermatozoa. They are not thickened or not swell, and the tail is straight. This is again the host reacted spermatozoa. This is the host positive spermatozoa. Clearly, you can see there is a swelling of the spermatozoa and also the coiling of the tail. Then after that, these are the some of the routine tests which are done at our lab also, and mostly the semen stations. Now there is again this is innovation is there in this semen evaluation method, and this CASA is available. This CASA is a computer-aided sperm analysis technology, which is used to evaluate the semen in very objective manner. So I will not go in detail because there is one lecture is scheduled about this specific applications of the CASA. So CASA nothing is a high resolution camera attached to the microscope, which takes the multiple pictures at a time and converts them into the video. And this video examines through the software with respect to the sperm number and motion characters within the frame. So this is a setup of the CASA. We have to feed the values that what should be the values for all this progressive SPR, progressive average path velocity, and again the morphological parameters are to be added. Then this is the motion pictures of the CASA. At a single time, multiple snapshots are taken by the computer, and it programs it into the form of the video. And monitor each and every spermatozoa, their kinematics, that there is their motion, or there is average jet velocity, what is their curvilinear velocity, velocities, and also the morphometry of all the spermatozoas in the field. These are the morphometric parameters. For example, what is the head length, what is the head width, the elongation of the head parameter, head area total, tail length, and these are the kinematic measurements which are given. So this is in the form of, then it gives in the form of the semen report, which can be used for further references. That is the total number of count were 116. The static were 30%. The progressively motile spermatozoa were 44, and motile spermatozoa were 70%. The slow were only 0.9%. And again, the morphometry of the spermatozoa which are evaluated. These are the kinematics output with the straight line velocity. It measures average path velocity. It measures, this is a curvilinear. This is the curvilinear velocity. This is the progressive velocity. This is the average path velocity which a sperm head is covering during its motion. Then some of the biochemical tests which are not routinely used in instrumentation, but we can use this for further references, or for if there is a problematic, if there is an infertility in a specific bull, or we can also use them for the research purpose. They include this pH estimation, methylene butyrate test, this physiologic index. This all tests have a common principle that they measures the metabolic activity of the spermatozoa which are present in the semen, and they digest to the a particular dye and gives the coloration. And again, resistance to environment. Here, the spermatozoa are exposed or given the stress, environmental stress, by giving them low temperature, low exposure, low temperature exposure, or high temperature exposure. And again, some biological tests or some of the in vitro fertility tests which are used for the research purpose. So one is this zona-free hamster egg penetration test, where this zona-free eggs collected from the hamsters are used to detect the fertilization rate, or the amount of spermatozoa which are having the capacity to fertilize. This is zona binding assay. Again, a zona OM is used for in vitro fertilization test. Now, this was all about the routine methods. Now, there's a high evolution, and day to day there is an improvement, and there is a research in this semen industry, and particularly on the spermatozoa. And there is a quantum shift of the methods which are used for the semen analysis. So routinely or previously, as we were using this mass activity, individual motility, viability, acrosomal integrity, concentration as the parameters for evaluating the semen. Now, the detailed kinematics, the host, DCPMT test, this incubation test, the few functional characteristics were used. And nowadays, almost every sperm functions are evaluated by using this CASA and flow cytometry for evaluating each and every aspects of the spermatozoa and the semen. These quantifications of the subpopulation, these maturation changes are also studied in some of the patients. So, for example, this is one of the tests where the capacitation status is estimated by using this fluorescent dye. This is non-capacitated acrosomal intact spermatozoa with illuminating hole over the head and the midpiece and the tail. This is a capacitated cell, again with the no fluorescence over the acrosomal region, because there is a loss of the acrosome. And this is again, this is capacitated some with the acrosomal intact spermatozoa. This second one, and this ER is a ecosome reacted sperm and capacitated sperm, so it does not show any illumination in the head, but the midpiece is showing illumination. Then nowadays, these are all the parameters or with the development of proteomics and molecular development, which are used for the evaluation of semen, that the spermatozoa should be highly motile and should be viable. The chromatin should be intact. The plasma membrane should be intact. They should go capacitation timely, and also the chromosome reaction. The membrane potential, mitochondrial membrane potential is detected and it should be high. The apoptosis should be reduced, low lipid peroxidation, low membrane lipid scrambling of the spermatozoa, and a low DNA fragmentation index should be there, so that the sperm can be given superior grade. So these are some of the sterilization or the routine procedures which are carried at our semen station. The sterilization are maybe can be physically by physical method. There's a hot air oven. Normally the glasswares and others are the sterilized using this hot air oven at -160 degrees Celsius for one hour. Then this rubberwares and other buffers and metallic instruments are autoclave at 10 pound pressure for 15 to 20 minutes, and the straws and seed pits and other biochemical materials or diluters, they are sterilized using the ultraviolet rays. So this is a wide mouth container with the frozen semen. This is a storage unit where the cryopreserved semen is kept for 45 days before dispatching in the field. These are the jumbo biological application containers. This is a container which is used at the field level. Now I will show you some of the aspects of this artificial insemination. How can we perform the artificial insemination? See, mainly at a time, one straw is to be collected. One straw should be identified and should be picked up from the container. And this canister should not be lifted above the level of neck of the container. Then identified straws is removed, and throwing is to be done at 37 degrees Celsius for 30 seconds. This is the laboratory end. Then the straw should be wiped dry with the use of the paper towel or the the napkin. Then all the details which are given on the straws are to be noted down. So normally the details are what is the identification number or what is the bull name, then it's where it is from. For example, our bull detail we can have is Mohan. It is from Gir. The what is the breed of the bull and what is the dams lactation yield are to be incorporated on the straws. Nowadays, barcoded straws are also available, which can be used for identification. Then we have to cut the laboratory end at a 45-degree angle, and the cut should be very clean. And then the straw is loaded into the artificial insemination gun by pulling the plunger. Then hold the gun properly. This is the plunger of the artificial insemination gun. Then the AI technique. So here's first the restraining of the animal is very much important. It should be restrained, and the glove hand is inserted into the rectum to palpate the reproductive organs. Then we have to manipulate the tract with your left hand as the rumen displaces the reproductive tract to the right. Normally, this cervix is at the near the floor of the pelvic cavity and anterior to the pelvic bone. Then the vulval lips are parted and the gun is introduced gently to the cervix under the control of the other hand, which is inserted into the rectum. And we have to hold like this and grasp the cervix and push it forward so that the vaginal folds get straightened and there is no chance of engaging your artificial insemination gun into the fornix vagina. And grasp the external opening of the cervix with the top on with the thumb on top and four fingers underneath to close the fornix and guide the gun accordingly into the cervix. Then the cervix should to be twisted and so that The gun is introduced through the cervical folds, and use your index finger to check the gun placement, that whether it is in the uterine body or it is in the in the cervix. So it should be 1/4 inch past the end of the cervix before depositing the semen, and push the plunger slowly so that the drops of the semen fall directly into the uterine body. And with the proper artificial insemination technique and placement of the gun, semen will automatically be deposited into the uterine body, and by the contractions of the uterine muscles, it will transport to both the horns and ovary. And if the gun sometimes we insert deep into the uterus or the uterine body, then it is inserted 1 inch through the cervix, then all the semen will be deposited only into the one horn, which is shown in this picture. And make sure to fully push it with the plunger and do not pull back, or otherwise the semen will be deposited into the cervix and the vaginal folds. And after the withdrawal of the gun, the vulva may be massaged to assist the sperm transport, and the clitoral massage gives the animal the sexual satisfaction of the natural service. These are some of the points to be remembered: that be gentle, don't use too much force while performing the artificial insemination. It may injure the reproductive tract, mainly the or cervix of the female animal. And insemination is a two-way step process. Set the gun to the cervix and then place the cervix over the gun. Deposit the semen just through the cervix into the uterine body. It should be done as quickly as possible, but not hurriedly or rapidly. So the sperm then hopefully reach and fertilize the egg, resulting in the conception and eventually a fetus. So if all goes well and the female animal becomes pregnant and carries the baby to term, then there will be a successful parturition and baby will be born. Thank you for your kind attention. Thank you.
Thank you very much, Dr. G.B. Solanki sir. Very nice. Beginning from the semen collection to evolution, routing methods and advanced methods, and at the last you have also mentioned how to AI and what are the care to be taken in the field during the AI. So it was nice presentation given by Solanki sir, and it is very fruitful to all the students, particularly for the UG students. Some questions are from students. The question is from Javed Khan. What is the normal reaction time in Gir and Jafrawati bull?
The reaction time may vary from individual bulls to bulls. On an average, it is 1.5 to 2 minutes in the Gir bulls, and it is 3 minutes in the Jafrabadi bulls.
Sir, is it vary in different species or in different breeds?
Yes, sir. Normally, this milk-purpose breed are sluggish. It is found that in the research that they are sluggish and they are shy in the sexual behavior. And this draft-purpose breeds, for example, Kankraj, they are very much furious and they are active in the sexual breeding also. So the reaction time is low as compared to the other breeds.
Okay, sir. One more question is from the student side. Sir, what is the cost of CASA?
CASA, there are different types of CASA. And nowadays, this CASA is available at 4 to 5 lakhs with some of these updations are available. But normally, this CASA is not used for the routine evaluation, because ultimately CASA is what you feed is it gives the result. If you change the standardized data over the this machine, then the results will vary. For example, if you feed in there that 40% is to be kept cut off, 40% post-mortality is to be kept for this semen straws for passing of that straw, then it will give you result that yes, the semen straws are good and the spermatozoa are highly motile. So again, it is around 4 to 5 lakhs. It is available and mainly used for research purpose.
Hi, sir. This is Dr. Dilip.
Okay, sir. how are you, sir?
Fine, sir. How are you, sir? Need to focus a little bit more on this word CASA. Sir
Yes,
is
sir.
saying that it is 4 to 5 lakhs. This is only for this motility parameters. Usually a standard CASA cost around 35 onwards, 35 lakhs onward.
Yes, sir. Yes.
And these are totally need based instruments depending on the user, what he wants. Because nowadays whatever the CASA are coming, they are not only confined for motion or kinetic analysis. They are measuring the morphology. They are also measuring DNA fragmentation. If they are having the fluorescent attachments, then they are going for viability, mitochondria, acrosome. So many aspects of the CASA are nowadays coming. And if you start from an integrated system, what sir was showing, that is IVOS 2 from the IMB technologies or SCARSCOPE from the micro-optics, they are costing around 55 lakhs with all loadings.
Yes.
And if you are going for an open system where you are having the flexibility of selection of the system or addition, then it is around 35 to 45 lakhs of rupees. Basically, the cost is of the high-end camera. The camera is costing around 8 to 10 lakhs of rupees, and the software is a patent. That software totally depends on the company, and they are giving us at 2,000 euro, 3,000 euro, because they are the supremos in that area. An Indian system is coming from the BioVis. They are charging hardly 10 lakhs or 15 lakhs, and they are giving the CASA. So it solely depends on your use, sir. I am also having a CASA from Minitube that is costing around 45 lakhs of rupees. So it varies. Regarding the use of CASA, yes, it is primarily we are using for the research purpose. And one should not fix the cutoff value. When I will give my presentation, I will tell you what should be the standard guidelines. Because in India, we do not follow the standard guidelines of using a computer-assisted semen analyzer. And that's why we are giving very difficult results, which are very difficult to interpret as well. So that's why it requires standardization, optimization, and as per your requirement, it should be fixed. And the rest of the things we'll discuss on 4th or 3rd, probably when I will going to present. I will go step by step. Thank you so much, Solanki sir. I heard you. It was a very nice presentation, very enlightening. Students should be benefited. Thank you so much, sir.
Thank you, sir. Thank you, Dr. Dilip, for adding information and enlightening us. Thank you.
Yeah, thank you, Dr. Dilip. We are also eager to see you on 5th day. Yeah.
Yes, sir.
So,
Yes, sir. Thank you.
Gajendra sir, one more question is from the student side is there. What are the different preference or preferred method of semen evaluation in the field condition? Or rather, if I say in the field condition, can we have any evaluation method to judge the semen quality? Solanki sir, I repeat the question.
Yes, please.
I repeat the question. What are the preferred method of semen evaluation in the field condition?
So sorry, the voice disturbance is there.
What are the preferred method of semen evaluation in the field condition? Or is there any techniques so we can judge the semen in the field condition, sir?
No, sir, there is no such technique available, but this small microscope which are developed for the field purpose, they can be used to evaluate this post-motility of the semen.
Okay, sir. So any more questions from the student side? If anybody want to ask any queries, so here we have two andrologists, Dr. Gajendra and Dr. Dilip Swain. Okay, so if we don't have any queries, then we conclude the day one of this vocational training. Thank you very much. Here, Dr. Wala is there, Dr. Sharma is there. Then I'm very thankful to Dr. Wasawa, Dr. Golkataria, then Dr. Sebaria is there, also Dr. Edi Patel, and Dr. Alawat is also joining us with us. Also, I am very thankful to Upadhyay Bhai, also is with to help our different technical session. And tomorrow we have a very great eminent speakers will be with us. Dr. G. Taru Sharma, she is a principal scientist and head at the Physiology and Gynecology division at IAI, and Dr. A.J. Damisar, a professor and head of Gynecology department, Anand Agriculture University. So we will be back at the 10 sharp AM, and we will start our two lectures from the eminent speakers. And afternoon will be one demonstration practical is there also. So up to that, thank you very much, everybody, all the participants. Thank you very much for being with us. And also for the student, we have sent one feedback form in the YouTube and as well as in the Facebook and through the mail also. So just fill it the feedback form and just say the how you enjoyed this first day of the vocational training. Thank you very much. Thank you.
So before concluding, I want to add some remarks. First of all, I would like to thank all the participants and particularly the different faculties who are joined over here through the Zoom meeting or YouTube or Facebook link. And we are very eager to have your feedbacks and your questions or queries, if any. And regarding the feedback, Dr. Padola very well narrated that after this session, every day we will send you a link, Google Form link to fill, and it is to be filled till the 9 every day night. So your feedback, your valuable suggestions are cordially invited, and particularly Dr. Madan. Dr. Madan also queried that if in his lecture, if you feel any queries, any feedback, it will be highly solicited by sir, and all the queries we will send to Dr. Madan. So please, it is requested to all of you, all the participants, please do fill your feedback form. And we are highly looking forward for the day 2 session. Thank you very much. Yes.